introduce our guest, the first speaker of today's webinar, Dr. T. A. Pawan Kumar, Senior Scientist of IMMT. Now I request Dr. C.H. Murali Krishna to introduce the speaker to the participants. Thank you.
Thank you, sir.
Thank you. Please,
So,
Murali
good
Krishna.
morning to all. It's my privilege to introduce Dr. T. Pawan Kumar Garu. Currently, he is working as Senior Scientist, CSIR, Institute of Minerals and Materials Technology, IIMT Bhubaneswar. And he received his PhD in Synthetic Organic Chemistry from CSIR, IICT Hyderabad in 2011 under the supervision of Dr. S. Chandrasekhar. So he got patents in 2014, Nalsar, Hyderabad, and also IP scientist at Dolcera, an IP firm. And he is the Inspire faculty in between year 2012 to 17, CSIR IICT Hyderabad. He did his postdoc with Professor Mukund Sibi, NDSU, Fargo, USA in 2015. And also he is the manager, IP and TT, NRDC, New Delhi in 2016. In 2018 to 19, he's the assistant professor, TGASC, Puducherry. So he got many awards. Some of them I reveal here. In 2018, he is the member Global Intellectual Property Convention, GIPC. In 2016, WIPO IP Patent Scholarship, and he got very prestigious awards in SEDB Young Scientist and also Telangana Young Scientist. In 2012, he's a DST Inspire faculty. And also in MSc level, he got 4 gold medals. And also he got university, he secured university first rank. So that is the command. And also he got 18 patents, 21 publications, and total citations are 333, H index 11, I index 16. And also he appointed several board editorial and advisory board member. His letters in organic chemistry, current organic chemistry, letters in drug design and discover, and also current organic catalysis and current organic synthesis. So these are the few lines about Dr. T. Pawan Kumar Garu. And with these few lines, I invite Dr. T. Pawan Kumar Garu sharing your views with us. Thank you, sir. And also I thank Dr. B. Jagan Mohan Reddy Garu, convener of this FDP, giving this opportunity. Thank you, sir.
Dr. Pawanji, please go ahead with your presentation.
Thank you all. Very good morning. And first of all, I thank all the organizers for inviting me to this session. And personally, I also thank Dr. Jagan Mohan Reddy Garu, who was a very good friend of mine. And we have been associated on several occasions. And I think now in this session, I'll try to take you to a different journey wherein how a chemistry work can lead to some patents. Especially most of the times we all look for the publications, research works leading to publications and some other things. But however, if we can put little care and fine tune our works, then those works can lead to new inventions and which will bring us some patents. So therefore, I have titled the lecture as Strategies for Protection of Chemical Inventions, a Researcher's Approach. And I think now I'll be sharing my screen. Hope you all could see my screen. So the title of the lecture is Strategies for Protection of Chemical Inventions, a Researcher's Approach. And if you look into this, this is my brief background. Of course, organizers have introduced me, but however, these are the different places, which is a blend of chemistry research followed by extensive research as well as with a parallel experience of intellectual property and technology development. Then these are some of my what you can say experience results out of my experience or my accomplishments. I have about 21 papers, around 19 patents. That is what is the major thing to be all participants to be looked into. What is the difference between having just publications and also having patents? So therefore, we have tuned most of our research works into such a direction that they will result into inventions, and further we will get some patents. And very recently, maybe around 2-3 days back, one of our Indian patent was granted. Those things I'll be showing here. And the basis of my presentation is this one. If you look into this, India amongst the worst on intellectual property index, and we are a country with 130 crore population. We have huge knowledge, we have huge tradition, and almost all 130 crore people are very, very intelligent, very, very creative. But however, where are we lacking? But the global statistics says that India is worst among intellectual property index. And this is what has put a major concern on me after my PhD. And unknowingly, I joined in a IP company where I could, and then from that, I could realize the importance of intellectual property for a practicing researcher or any academician. And from that time onwards, I tried to explore IP as a parallel career. Then, so when we talk about science, science can lead to discoveries, science can lead to inventions. And most of us, we struggle in finding the difference between what is called discovery and what is called invention. So therefore, I try to simplify this. Discovery is something which is already there, but however, you are the first person to show it to the world. But invention is not like that. Invention comes when there is a problem. So invention can be termed as a problem solution approach. Therefore, see this, Eureka. Whenever we look into this picture, Eureka strikes to our mind. Eureka, Eureka. So therefore, that is a discovery. And instead of him, if instead of Archimedes, if we see it or if someone else see it, the same thing will happen. But we did not observe, he has observed. Then, so when we talk about inventions and patents, there is something called as claims. Claims are nothing but the boundary of an invention. Like you have boundary for your property, claims will form the boundary of your invention, and therefore there are different kinds of inventions possible. Similarly, those inventions can be protected in the form of different claims, and especially here I am showing some types of claims which are more relevant to the chemistry people or chemical people with chemical sciences background or chemical engineering also. So the first one is product claims. Whenever you develop a new product, you can go for a product claim. Then process claims. So these claims or this protection is towards your process if it is new. And the third one is markage claims, especially for organic chemistry people. Where do we generate a large number of analogs, similar analogs? Here I tried to show you what is called a analog chemistry. So if you replace chloro with halogen or any methyl or ethyl with alkyl group, so those structures will become very generic. And such kind of representation is called as a Marquis representation, and those claims are called as Marquis claims. And there are some claims called as gypsum claims, and also the other claims are Swiss claims. Swiss type of claims is you have a product, maybe some drug molecule that is used for some treatment. Now, if the same molecule is found for some other disease, that is the second use. And if it is for this is third use. So any drug molecule, if అదే మా దగ్గర ఇక్కడ ఫుల్ వర్షం ఉంది, దానికి కొంచెం ఇదైనట్టుంది. సో ఓకే, ఇట్ కెన్ బి కవర్డ్. ఓకే ఓకే ఓకే, సరే సరే, ఓకేనా. So these are the claims that are allowed. Now let us go to the next slide. So from here onwards, I try to show you some of our research works which led to inventions followed by patents. And this is a work on 3-hydroxyproline, which is available in nature, but however, its availability is very rare. So therefore, we tried to develop a synthetic strategy wherein we could synthesize all possible isomers, all possible stereoisomers, because it has 2 chiral centers. So 4 stereoisomers are possible, all 4 we could make, and with the simple steps. But however, we could only end up with publication. And then this is another example where we have utilized simple click chemistry to see all the compounds here, amino triazoles or triazole amino oxides. All these are reported for the first time in literature. For the first time, they have reported in the literature, and by that time, we could realize the importance of IP, and we have filed a patent first, followed by publication. And all these compounds were tested for different biological activities, but however, we could not find a very prominent activity. But all these compounds are new. And this is the another work. It's already a granted patent, which talks about an antioxidant compound having anti-atherosclerotic effect. Here we tried to synthesize a eskellatin derivative, a very simple molecule, and it also involves very simple chemistry. But however, designing that structure followed by its biological activity analysis was very prominent, and we could get 2 US patents, and the recent Indian patent we got granted for this one. And there is a one United States patent, United Kingdom patent. And this was our very initial work, and in the year 2019, these patents were out-licensed to Sun Pharma. They are working on that one, and it was a deal. I think most of you might be knowing. There was a news from CSR. IACT has transferred its technology to Sun Pharma by patent out-licensing, and which could be estimated to the extent of 240 crores in coming years. But however, as a first line payment, IACT has received about 5 crores last year. So that is one of the success where we could transform our basic research work towards the development of a invention and leading to further applications. And this is how we made. This is the structure of eskellatin. But of course, I think it is very dim. I can understand, but I tried to show you the patent itself here. So this is what everything will come into the patent. And after the patent, we also published it in other journals. And it's a very simple molecule. Only 3 or 4 steps was required to make this molecule. But however, since we realized the importance of IP, we could push it towards patenting, and then all the other stories have come into picture. Now Sun Pharma is working very seriously on this, and if everything goes well, within about 3 to 4 years, this may become a very good drug candidate also. Then this is another work which talks about pyroxyindole compounds as GSK3B inhibitors. All the structures are newly reported, but however, the strategy, chemical strategy used, it involves different types. Allylation, Bach protection, pyroxy ring, and 5-membered as well as azide. We could able to do click chemistry over that one. Then the second amine was replaced by some other groups. So therefore, we could able to design around making several analogs, and all this analog library was tested for GSK3B inhibition property. And we could find very prominent results, and very recently we got a US patent also for this one, and a PCT has been applied, and Indian patent is under examination. And these are all the different analogs of this compound based on the click chemistry, very very simple click chemistry, which could by which we could generate about 50 or 60 different analogs by various structural modifications, and at the same time several uh, amines were also prepared, cyclic amines. So all these were tested for this, and a very good biology is going on. Then this is about a process. So those two are examples of the product patents. So two new products were made, and then the biological activity was tested. And this is an example of nicotine, which is a process patent. Nicotine, generally, when we talk about nicotine, people have a very negative feeling. Those who drink tea, those who smoke, all that contains nicotine. So therefore, that is not good. But however, nicotine as such is also having some good properties, but in small quantities. But however, when we observed the naturally available nicotine, always it is contaminated with some or other impurities. By whatever so method you try to purify, those impurities are not going out. Then we thought, why can't we put some efforts to synthesize the nicotine? So therefore, we started synthesis, and I personally made the literature survey, and if we could find about more than 100 or 200 patents on this for different processes. Then based on so much of efforts, we have come up with this scheme where in within 3 steps we can make the nicotine. And however, this nicotine preparation can also be made in one pot operation. So stepwise we can make, and one pot also we can make. And that is the reason why we thought this has a lot of potential in coming days, and we have gone for patent application in India as well as PCT and Europe. And very recently, EP patent was granted, and Indian patent is under consideration. So and many discussions are going with several clients, wherein we are trying to transfer this developed process to them so that they can take up a large pool of process patent on alpha lipoic acid. Alpha lipoic acid is found to be a very prominent compound, and this is an approved drug for some kind of diabetes. It's an antioxidant and also a dietary supplement. And here I want to insist that if you visit any doctor, please carefully observe his table, his or her table. On his or her table, you will find some medicine which will definitely contain alpha lipoic acid. So therefore, its utility is very high. But however, we found that in India, no one is making this lipoic acid. We are importing 96% of the imports are from China, 4% import is from America. And we need about 410 tons per year. And when we need such a huge quantity and we don't have any process, then we thought we should look upon this one. Then we tried, and there were several processes reported. Whatever alternate retrosynthesis we do, either it is already existing or already otherwise it is very similar to something existing. Then we thought maybe it is very difficult to come up with a completely new process, but however, and finally we could end up with an improved version wherein with a good efficiency, and we can get this product in higher yields and higher purities. So therefore, we coined it as a start and end protocol, one pot operational protocol. And instead of using several solvents, we could develop this process in a single solvent system with a very operational feasibility and simplicity. And finally, we got a patent, and also parallelly, we are trying for continuous flow method also. This technology is highly economical and highly viable. And very recently, the discussion was going on with Sami Labs in Bengaluru, and maybe in another month or so, this technology will be transferred to them so that India can make lipoic acid on its own. And so that is about another process patent. This one is, it is like a product come process patent. Zafirlicast is a drug molecule which is already in use for treatment of asthma. many companies are making this one. But however, most of the existing processes are lengthy, time taking, and they are producing lower yields. We thought nowadays మళ్ళీ అదే వచ్చేసిన ఎందుకంటే వర్షం ఉంది కొంచెం వచ్చేసిన So this is about Zafurlic acid. So we have generated about 40 or 50 different unlocks for each and every step wherever I am showing a generic structure. The structures where you do not find any specific functional group is called as a marked structure or a generic structure. And we could able to develop a process for this molecule which contains only 5 steps. A 5-step process, and as of now it is the very shortest reported process for this molecule. And we have filed an Indian patent and US patent as well as a PCT application. And this one is another example of again a product come a process patent. Nimbolide. Nimbolide is a natural product which is found to have very good anti-cancer activity. And being in IICT, we thought IICT is very famous for natural product chemistry, and India as such has got so many traditional plants wherein we could obtain very important biologically active natural products. And we found nimbolide was such a compound wherein we can design around to generate more number of analogs, and those new analogs will become new products. The reaction steps that we use to make these products will become a new process. So therefore, we have generated about 15-20 different analogs by using Diels-Alder reaction and several other reactions. Here, this is the left side one is the generic structure. The right side one are some of the examples of the analogs that we have prepared. And now we got a chance to make an extensive biological study of the nimbolide family of compounds. And based on this work, the scientist who involved in doing biology, his name is Dr. Ummani Ramesh. He is in IICT Hyderabad. Now he got a project, 1 crore project from CSIR to do extensive biological activities, because out of the analogs generated, one of the analog was very, very effective. It is showing biological activity at nanomolar level. So therefore, there is a big scope to come up with a new molecule with good biological activities. And maybe in near future, we may also tie with some of the big pharma industries so that they can do for clinical trial. All those steps will come in near future. So for this, we got an Indian patent and a US patent as well as a PCT application. And this is another example of indole sulfamyl hydroxybenzamide derivatives. Again, this work can be classified as a product come process work. We have developed all the developed compounds are reported for the first time in the literature. The process we use for developing these new molecules, maybe if you look into individual step, that kind of step might be already known in the chemistry. But however, applying known sequence of steps to make a new molecule will also become a new process. And if you do any improvement over the existing process, that will become an improved process. So therefore, with a combination of all these strategies, we could able to prepare a very huge library. Almost we made around 108 compounds or even more. And all these compounds were tested for HDAC inhibition activity. And in literature, we could find that compounds showing selective HDAC inhibition activity are very, very rare. It is not that easy to find compounds with this kind of activity. Then we thought we should explore them even more with even more effectiveness. So therefore, we have designed around, again developed another 100 compounds. There are about 200 compounds. Library is there, and extensive studies are going on. And as of now, the present status is they are showing very, very good activities. Meanwhile, we could able to file Indian patent, PCT, and in near future we may also go for patenting this process or product in several other countries based on the biology. So here, whenever you look into the structures looks to be very simple. Chemistry looks to be very simple, but however, it has a very, very great application. And this is another example of isothiocyanate compounds as SMAR1 stabilizers. If you look into this one, I have represented this as A, B, R1, R2, R3, R4, and NCS. NCS is isothiocyanate group, and that whatever A, B, R1 that represents the different skeleton. And these are some of the examples which I have shown on right side. We have made a library of compounds again. I think by this time you might be knowing our strategy of developing new molecules and taking them for biological applications, showing their biological activity. And if they are found to have very good biological activity, we are going for patents, because we get patents as well as we get publications. So therefore, we got an Indian patent for this, US patent, and very recently, first examination report of Indian patent for this has come, and we have replied to that one. Maybe in another 6 months, this patent will be granted. And this compound is also showing a very, very good level of SMAR1 stabilization property. And this work has been recently published in Antiviral Research. With this, we have done in collaboration with NCCS Pune and CDFD Hyderabad. So here, this slide is on the basic research, which involves development of new organocatalysts and their application for asymmetric transformations. We used to work on proline catalysis as well as multifunctional catalysis. Here, I try to show you in a very short manner what are all the different kinds of triazoles or different proline analogs we made. We have made a large number of catalysts. Many of them have already been published, and for something, still the research is going on. And the other one is AZT thiourea analogs. We try to design these catalysts with new combinations so that we are expecting a good amount of selectivity in the asymmetric transformations. These are some of the examples, and we found that venlafaxine and desvenlafaxine, these two are the drugs. But however, when we have examined them carefully, the very simple Michael reactions that we do, whenever you look into the literature of organocatalysis, Aldol reaction, Michael reaction, Bailey-Sielmann reaction, there are 4-5 reactions which are called as benchtop reactions. Whoever does a new catalyst, initially they will test with these reactions and further they will expand. So therefore, we thought with this simple Michael addition reactions, we can able to make these compounds. But however, the more important point is we have to get very good level of selectivity. So therefore, with all our developed newly designed catalyst, we have screened that, and we found one of our catalyst was very prominent in the strategy of making this molecule, and the process involved around 6 steps to complete the molecule with good level of selectivity. And later on, we could also generate large number of analogs for this molecule. And recently, we have been applied for Indian patent, and still some of the works are going on. Once that is completed, then we will be going for publication. And if it really gets success, this will become one of the shortest route for making of this molecule, and it will also give us the possibility of making several analogs, because with Michael addition, as the as we change the nitrostyrene substrate, we can get those many number of unlocks. So therefore, there is a possibility of generation of a big library for this method. Generally, as a chemist, we have to play a very big role if we feel that we result into proper output. But if a chemist collaborates with a biologist, if a chemist collaborates with a material scientist, or if a chemist collaborates with another person who is from any other discipline, we can able to generate a very output-oriented, very productive work. In similar lines, CCMB was looking for someone who could provide them with some artificial nucleotides. So this is the target compound represents the artificial nucleotides that CCMB was looking for. They wanted to study some proofreading mechanisms in tRNA and amino acids linkage. It is like transcription process. So therefore, when they have approached us, initially we were hesitating because this kind of chemistry is very new for us. But later 4 years to come up with the molecules. So this is the targets, but we told them we cannot generate compounds. That would be a very good sort of view. Then this is what they were looking, and initially we have proposed these two things. This is what whenever they are doing. So so while binding, what happens is generally when it binds or when it recognizes, it will not make any mistake. But however, there is a 1% chance, not even 1%, even 0.1% chance of doing some mistake. Our CCMB biology people wanted to study in what way it can do mistakes. So therefore, we started developing this. And this is the synthetic strategy for making these nucleotides. But of course, I can understand it is very congested here, and it was very complex. And every step we have to purify through preparative HPLC only, not by regular column chromatography or any other methods. All these compounds were very polar in nature, and isolation was very difficult. Even characterization is even more difficult. But however, after 3 or 4 years of continuous efforts, we could able to give them 3 compounds with 10 milligrams each. So now their biology work is going on, and it may result in very good output in coming years. And this slide I am showing you because whatever I spoke all through this session is mostly related to converting our research results into inventions, then taking them for patents, obtaining some patents, then talking to the industry or talking to some other people so that we can explore them to the further extent. Because we all know about the wars that have happened between different regions to capture the territories, Alexander the Great or several other wars. But however, in future or in coming days, the ownership or the wars will not be on the territories, but it will be on ownership and exploitation of ideas. The Western world is doing to a larger extent to protect their properties, which are the properties of their intellectual creation. But however, in India, we do generate a lot of things intellectually, but we are failed in protecting them and we are failed in exploiting them. That is the reason why still we are called as a developing nation. Whereas the small countries like Japan, Korea, all those things, they were they were very disastrous. People have exploited them to a larger extent by World War or some other reasons. Even then, they could able to cope up and they could emerge as a developed nations. But however, when it comes to our country, we see only Indians only gave to the world. But what happened now? What happened to us now? Why do we get worst in intellectual property index? If we are so intelligent, so creative, we should have had a very good stand. So therefore, future wars will be over the exploitation of ideas or intellectual creations, not just for territories. So therefore, if we are formulating hypotheses, accumulating data, and doing extensive studies, if this is the kind of research we are following, then this is the old way. Because this type of research can only give us good number of publications, good number of knowledge, good number of material. But this, if it is translated to output, productive, and if it goes to the market, then only we get lot of advantage. So therefore, to do that, one has to do the research, then it has to be taken for next level. Formulate a hypothesis, patent it, raise some $1 million. So finally, if we can make some money out of our inputs or of our research efforts, then that will lead to a successful model in the present global scenario. So therefore, if we can do this, this will become the new way of research. This is what is followed by the Western people. So here I try to show you in a very concise manner all our research efforts that we have discussed. This is about organocatalysis and different works. Then these are about all the collaborative works. First one is spiroxyindole leading to GSK3B inhibitors. In collaboration with CSR IST, we got about 4 patents. The second one is indolyl sulfonamide derivatives as HDOC inhibitors, that we got about 2 patents. The isothiocyano compounds we got that is in collaboration with NCCS, and this is with CCMB for recognition of the tRNA proofreading mechanism. And this is nimbolide analogs. The other one is nicotine process. It's a process patent. And the bottom one is TPP coupled escalation analog, for which we got 4 branded patents, and recently the same one. It got a patent, the patent was out licensed to Sun Pharma, and about 5 crores were already received as first milestone payment by IICT, and we are expecting this will become a very good success in coming years. So therefore, my the main essence of making this kind of presentation is to show you that we have a very good level of knowledge, we have high level of experience, and we have high level of creativity. But the thing is, we are unable to connect them together. If we can connect our knowledge, experience, and creativity, certainly we can do wonders. And still again, I can say that in coming years, we can lead the world. So therefore, for an effective OIP outcome, connectivity between all these things are very, very important. And as an individual, it is our duty to promote, protect, and profit for whatever works we do. And for that one, we need to have proper awareness, understanding, and then we need to have the contributing attitude. Once we can do this, then certainly we can emerge as a very, very strong nation, as a nation, and also as an individual. Therefore, apart from our chemistry research, I will also I also have experience in IP patenting aspects, and parallelly I also do this research, search and analytics of patents, writing patents to several people in different domains, searching of patent literature or prayer arts and all these things by using several databases. And we do conduct outreach activities, and also I have drafted about 30 or 40 35 patents till now, and I wrote patents to Professor Govardhan Mehta, and even in ISCT, when I was in IST, I think I wrote about maybe 20, 25 patents in chemistry, chemical engineering, and several other domains. And I also involved in promoting IP by conducting awareness sessions, conducting classes for PhD students and PG students, as well as workshops for the faculties and all these things. So here in a single slide, I could able to show you or give you the picture about what is called intellectual property, why it is so important for a researcher. Then as an institute, or maybe it is a college or a university or any organization, we should have a strategy for aligning our capabilities. But however, in India, we are failing to do so. Whenever we do some R&D activity, that work has to be screened for patentability. If there is a potential for going for a patent, first of all, the institute or the research group should plan for filing the work for a patent, then go for publication. With the same work, you can end up with a patent as well as publication. But in most of the times, what do we do is whatever works we do, we just go for publications, then forget it. Then we plan for some other work. But however, if we can go for a patent followed by publication, it will be helpful for us in commercial exploitation, and all this activity will also bring us visibility and credibility. Of course, just with publication also you may get visibility and credibility. But however, if you have patents, if you exploit them commercially, that credibility and that visibility will be far higher than the regular visibility. So therefore, IP plays a key role in all functional stages for academics, research, each and every sector. And with this slide, I think I try to show you the importance of aligning research alignment of our research work with the IP activities, so that all the research that we do will lead to productive output. And I think I'll close here, and thank you all for, and I also request you all to give your feedback or any suggestions or inputs. And I think I have made justification for the title what I gave, and maybe I have opened up some new avenue for the researchers or the faculties to think upon. Thank you.
Thank you, Dr. Pawan Garu. Very good presentation. I think there is some problem from your end, some rainfall, as you said, rainfall is there, right?
Yeah. Actually, it is for us. It is some cyclonic effect is there, so there is some disturbance.
Disturbance, yeah, we understood. So no problem. So very good, Dr. Pawanji. You have given nice presentation, and in your presentation, you said that a new catalyst design, right? In one slide, you said that, right? Proline
What?
catalyst. Slide contains new catalyst design patents.
Yes.
Yeah. It is available commercially? We can purchase it, and what is the cost?
Most of the organocatalysts, if you
Okay.
try to purchase them, they are very costly.
Okay.
But
So we can
most
test.
of them you can make in the laboratory by simple strategies.
Oh, it is very easy to prepare in the laboratory, right?
Yes, it is very easy to prepare.
So by using those catalysts, we can test general basic reactions with very good yield. We'll get very good yield, or you will get optimized yield, or we have to develop the procedure.
Yeah, it is like this. Initially, you need to do some optimization studies.
Okay.
For that, you need to develop a library of organocatalysts. Then you have to go for optimization studies. You have to identify a very good catalyst. From there, you can explore your studies again.
Okay.
If you develop only one catalyst and work around, means maybe that may not make much sense, because that catalyst may be efficient or may not be efficient.
Okay. And coming to the other point, what you have mentioned in your lecture, and you said very good point, that collaboration of a chemistry, a person like chemist with other biotechnology or microbiology will definitely lead to the formation of good things. and we can produce very good publications. Very
Yes,
good point you have raised in your presentation.
because that is what we are lacking in India. We do
Yes,
mostly individual centric research, and finally, if you ask a chemist, most of our products or our research works, they will go into drain. That is the reason why there were several articles also. Most of the chemistry research is like a sink research.
okay. Yeah, yeah, I agree with your point. Very good.
Because if I make 1,000 compounds, if you ask me, can you give 10 compounds? Means I say no. Already I published. Now compounds are not available. Then why is that I am making them and only just publishing?
Yes, yes. Correct, correct. Correct point you have quoted in your presentation. I agree with your point also. Now I request any participant, if you have any question, please. I think there are two questions in the from for your presentation, Dr. Pawanji.
Yeah, please,
Uh, uh,
please you can ask any questions, but please be louder.
that question raised by one participant. Are you able to listen my voice now?
Yes, yes.
So the first question is there. You see at the questions and answers in box is there. Whatever we do research and invent new compounds, can we patent all these compounds? Is it newly novel and biologically active compounds only patentable? That is the question raised by P. Triveni from
Ah,
Vikramasingapura University in Nellore. Oh, she is working as assistant professor at
right, right.
Vikramasingapura University in Nellore.
I
Please,
will answer. I will answer.
please.
See, if your compounds are completely new means new. So, and if they have some application, either biological application or polymeric application or material application or any application, then you will definitely get a patent. But they have to be completely new.
Okay, okay, okay. Those compounds should be new,
Yes.
novel compounds, right?
Yes.
Yeah. There is another question from Pin, same Triveni. She is asking another question. She is raising
Uh,
another question. What is the minimum requirement of applying patents? She is asking.
minimum requirement of
What is the minimum requirement for applying patents? For applying patents.
patents. Actually, there is nothing like minimum requirement. If your work is novel, if your work is inventive, if it has inventive step or non-obvious, someone by looking at your work should not say that, oh, what is there in that one? It is very simple. That should not be there. And if it has some utility, if your work matches to these three criteria, then it can be patentable.
Okay, okay, okay. Very good. Any more questions from the participants? And another question is there, sir, from a lecturer from a college, affiliated college.
Okay.
The question is, class of compounds is required, or for single compounds is enough for patent?
Yeah,
Another Leela is
right.
asking, Miss Leela is
Sure.
asking.
See, you need not have class of compounds. A single compound is also enough to get a patent.
Okay, single compound is also
Yeah,
enough to
single
patent.
compound is also enough to get a patent.
Yeah, they got clarification. Any more question from the participants? You can have you can interact with Dr. Pawan Kumar. Pawan Kumar is very active and dynamic, and he is a very good worker. He is my good friend also.
So if you have any queries, you can ask now. Otherwise, at a later stage also, if you come upon any queries, you can just write to me by
Yes, yes,
email or through WhatsApp. Anything is fine. And the only intention is we have to contribute towards strong IP, because we are
yes.
doing big kind of research we are doing, but however we have to translate those research works into good IP also.
Yes, yes, I agree with your point, Dr. Pawan Kumar. So I think all our participants will contact you by email or phone so that they may get idea, and you please guide them for the benefit of their future, and whatever you said, that collaboration, and they will do in a very perfect way. Uh, Thank you, Dr. Pawanji,
Sure,
for
always welcome. Thank you.
Yeah, for spending your valuable time with us, even though you are busy with your schedule, you have accepted to deliver a talk in this faculty development program. Oh, thank you very much. And in future, we will meet soon at our university also. Thank you very much, Dr. Pawanji.
Thank you all. Thank you.
Now we have uh, another speaker. Today's speaker, we have another speaker with us, Dr. S.S.V. Ramasastri, who is working as an associate professor at ISER Mohali, located at Punjab. Yesterday, we have already introduced about the speaker to the participants, and there is some small interruption while he is giving the presentation. Now, now Dr. Ramasastri sir will come into the picture, and he will deliver about his talk. His talk is a new paradigm in the synthesis of privileged heterocycles. He can talk on this. Sir, Ramasastri Garu, are you there?
Yeah, I'm here.
Good, good morning. Good morning. Yeah. Good morning. Good morning. Yesterday, some problem is there. I think maybe some. Yeah. So.
To all the organizers, all the participants, I apologize for that.
Yeah.
It's unfortunate that I got disconnected.
Yeah, we waited for a time, and Professor Ramacharya also is trying to connect you. We called you several times.
Yeah,
So that we are not able to connect you. Anyhow, today our participants are eager to listen your lecture.
more eager actually.
More eager. So I think there is no introduce you, because already we have
Sure.
introduced you.
Yeah, we'll straight away get into the presentation.
Yeah.
Yes.
So now I request Dr. SSV Ramasastri Garu, who is working as associate professor at ISER Mohali. Uh, and one more point I want to tell with the participants. Dr. SSV Ramasastri is from Kakinada, which is a very good place in Andhra Pradesh, and it is located near to our Nanna University, that is at near to Rajamandri, Kakinada. He belongs to East Godavari district. So now I request Dr. S.S.V. Ramasastri, who is working as associate professor at ISER Mohali, to address our participants. Thank you, sir.
Thank you. Thank you very much. Yeah. First, let me ensure the connection and.
Okay.
Okay, you're able
Yeah.
to see.
Yeah. Yes. Yes. Just we are getting your and better to maximize or that
I'm doing it.
you put that
Yeah. Now
also.
is it fine?
Yeah. Yes. Yes. Yes.
Okay,
Yes.
thank you.
Thank you. Thank
Yeah,
you.
thank you very much. Let me get started. I think the title is something that I shared with all of you yesterday as well. This is a very general title, as told yesterday as well. I think I just initiated this talk. So it deals with primarily the synthesis of privileged heterocycles. As the name suggests, privileged in the context of medicinal chemistry or from the drug discovery point of view. So what our lab does at ISER Mohali is we try to develop different methods employing metal catalysis as well as metal-free chemistry, also as shown here. As of metal-free chemistry, we tend to employ phosphines as catalysts. And what are the methods that we develop? Ultimately, we intend to find applications in the form of total synthesis of natural products. So while these two areas are something that we actively pursue, for today I'm going to talk to you only about the metal chemistry, that is catalysis by transition metals. Especially, you will see that majority of the talk would be about palladium mediated chemistry. Okay, so as of this chemistry is concerned, within metal chemistry, we here today I'm going to talk to you about two concepts primarily. One is, as is shown, one part bi and trimetallic orthogonal tandem catalysis. As the name suggests, this involves the development of bimetallic as well as trimetallic catalysis. This terminology looks a little bit complex, but as you go through the work, you will realize that It's very simple. It's only about how we condense the concept in the form of this particular sentence. But otherwise, these one-part strategies will lead to the formation of the synthesis of compounds such as cyclopentane fused indoles, beta carbonyls, fused pyridines, and also I'll talk about the mechanistic details where appropriate. In another concept, I'll also talk about palladium catalyzed intramolecular allylic alkylation reactions. So this other way you can also understand by textbook chemistry, as you refer to, it's otherwise called Suzy-Trost type chemistry. Yeah, so in this context, we have made some contributions as of the synthesis of cyclopentadienes, cyclopentene fused arenes and heteroarenes, carbazole synthesis, and otherwise very recently we have also demonstrated the application of this strategy in the synthesis of spiroxindoles, which are medicinally important and from the natural products point of view also they're very much relevant. So our contributions in these fields were recognized, and we were actually invited to compile our results in the form of reviews. These were the invited reviews by the editors of these respective journals. One may go through. It just not only compiles our own work, it also compiles the relevant works of other research groups as well. Okay, let me straight away get into the work. There's no great background for what I'm going to talk to you today. This is the one slide background I can tell you. As you see from this slide, the reaction of this benzothionyl carbinol with this 1,3-dicarbonyl was planned originally. So we intended to obtain this product. So we wanted to treat this compound and under acidic conditions react with this nucleophilic component, that is this 1,3-dicarbonyl, obtain this product. But it so happened, while we are screening different conditions, it so happened that this reaction didn't stop at this stage. It subsequently underwent intramolecular Friedel-Crafts type alkylation followed by dehydration to give these cyclopentene fused benzothiophenes. You also see that this is actually is a fully substituted cyclopentene fused benzothiophene. So the optimized system in our hands was it's a polyphosphoric acid, PPA, polyphosphoric acid. This is a one-part process. So you start from here, you end up getting this particular product. This has demonstrated fair scope as of the 1,3-dicarbonyls are concerned, or as of the groups such as R1 are concerned. You can employ aliphatic chains such as this, for example, or aromatic groups, or heteroaromatic groups, and even alkyl groups such as this. So this is actually the background. This I should tell you, this work literally changed the dynamics of our chemistry. You will see how it changed in the couple of slides coming by. So while this work was underway, we thought, why can't we do the same reaction on indonyl alcohols such as this? So can we get the same product, what we have obtained on benzothiophene? Can we get the same compound? Can we obtain the cyclopentene fused indoles, especially that these are fully substituted systems? Why? The primary reason is the relevance of indoles in the context of medicinal chemistry as well as natural products chemistry. One can find several natural products, bioactive natural products with this particular core. So I'm not showing that in the paucity of time, but otherwise, as the arrow shows here, we couldn't obtain the product. So we tried several conditions. However, this reaction was somehow not successful in our hands. We obtained a different product, which I'm not showing here, and we also published that work. But I'm not deviating from the discussion today. I'm moving forward. At this time, with an intention to obtain this compound, we came across this report by Chan. What Chan has published in 2010 is the reaction of this aminoinol in presence of gold catalyst and in the presence of an appropriate nucleophile obtained products of this kind, indolylic systems. So if you see the representative structures that are placed here, so one can see that a nucleophile such as methanol got introduced here, a nucleophile in the form of electron-rich arene, electron-rich heteroarene, and nucleophilic fluorine got introduced at this position, indicates to us that transiently a indolyle cationic equivalent got generated. So that's what we wanted. Obviously, as you see this reaction, when we treat this compound with acid, it undergoes a dehydrative formation of indolyle cationic system, or cationic equivalent, I should say. So similarly, Chan could be able to develop a procedure where indolyle cationic equivalent could be developed, but of course not employing acid. This is a Lewis acid, otherwise it's a gold complex. So the scope is fairly broad. One can go through this particular work, but to me, what is important is the preferential generation of indolyle cationic systems under even mild conditions than any acid that actually otherwise suitable for the generation of this kind of substrates. Okay, so with this concept, what we thought, we now combined, as you see in this particular scheme, we combine two concepts. One, Chan's concept. This is the Chan's concept. What Chan has done, aminoinol in terms of gold chloride, obtained this 5-exo-dig product, that is this indoline, and subsequently from here on we combined our own work, that is acidic treatment of indolines, leading to transient generation of indolyle cationic systems and subsequent entrapment of these kind of systems with 1,3-dicarbonyls now undergoes nucleophilic addition, subsequent intramolecular Federic type alkylation, and dehydration gives us this 1,2,3 trisubstituted cyclopentene fused indoles. The scope of this work is quite broad, as you see here. So variety of 1,3-dicarbonyls such as this can be employed in this work. Not only 1,3-diketones, 1,3-ketoesters, ketoamides also can be used to obtain respective products. You can also use a variety of R1 functionalities as of this work is concerned. You can see heteroarenes, arenes with electron-rich, electron-poor substituents. Even tetracycles of this kind can also be obtained with the appropriate planning of the respective substrate. So after working on this, so what came to our mind was, yes, okay, fine. As of this particular equation is concerned, yes, we can actually obtain indolyle cationic equivalents, but this happened in the previous work when R was either aryl group or an alkyl system. So what we wanted to do, can we introduce an alkynyl system, that is an olefinic system at the position of R? Then this actually represents a 4-pi electron system. If you see, this is a 4-pi electron system, which is very much favorable to undergo a Nazarov type cyclization, and subsequent deprotonation can give again cyclopentene fused indoles. This was a hypothesis. This was literally drawn on the piece of paper, and indeed we actually made this substrate to generate the respective indoline, but for this you need little modification of the starting compound. While we used aminoinol earlier, this is eninol. You can see in-ine-ol and amino group appended to the ortho position. Under this literally the same condition, that is gold catalyzed conditions, gives this indoline. Now subsequent treatment with chelating amount of triplic acid in situ must have generated this 4-pi electron system, Nazarov type cyclization. Provided access to the, now this is 1,2 disubstituted cyclopentene fused indoles. Okay, this is very briefly the scope I am presenting here. You can see varieties of in cyclopentene fused indoles. More interestingly, you can see I'm showing only 2 out of many other examples that were made as of these pentacycles are concerned. The pentacycles, I mean to say, of 5 ring containing compounds. So this is possible with planning of your precursor. So if this particular R3, R4 is cyclic, for example, or in this case it is cyclic, so then you can obtain compounds such as this. Otherwise, this can find, although we didn't explore, can find, I can tell you good applications from materials point of view. More interesting is the indole steroidal hybrid that you see here. As I said, mere planning of your precursor can lead to complex structures such as what you see here. This is, we can obviously call this as a modified steroid. In this case, it is a modified cholesterol. In this particular case, is a hybrid molecule of a heterocycle such as indole combined with a steroid. This is a more interesting case. The last example that I put up, I'm not showing the complete details of the synthesis leading to this particular molecule. Otherwise, this compound can be called, belongs to a class called polyvalent family of natural products. Polyvalent is a natural product containing methyl group at this position in the place of this hydrogen. But since our methodology cannot tolerate a methyl group, as you can understand in the precursor, we wanted to target NAR polyvalent. That's what you see here. We wanted to synthesize NAR polyvalent. NAR, obviously, as you all know, one carbon less to the primary carbon skeleton. So we prepared a compound up to this level with hydroxy protected, of course. However, several of our efforts to hydrogenate these two double bonds were unsuccessful. So we tried quite a bit of time to protect or hydrogenate this diene moiety, but it was not successful in our hands. So we published as such in this particular work. But otherwise, this represents the complete carbon framework. So of NAR polyvalent. So with this background, we also thought, what can we do something else with this concept? Can we exploit this in a way that we can also access biologically very much relevant targets such as these carbolines? In this particular instance, these are called tetrahydrobeta carbolines, as you see. This is tetrahydrobeta carbolin. Our intention was to obtain, start from here, in one pot, can we get this compound? Okay, so if you see this particular compound, this is a little bit modified compared to the earlier designs, if you see. So that is this amino enol now is having a allyl system introduced. Now it is a propyl alcohol, but it's a tertiary alcohol. Okay, now what is the hypothesis, or what is the idea when we actually initiated this work? Idea was that with the experience that we had earlier, we questioned whether this compound can be converted selectively to this particular indoline under catalytic systems such as whatever that can be relevant in this particular instance. Okay, can a catalyst C1 catalyze this system, this compound to this indoline? Then can now introduce amine leading to the formation of this indolyl amine. Now, can I identify another catalyst which can actually accomplish intramolecular hydroamination leading to the formation of tetrahydrobeta carbolin? Intention, as I said, to do all this in one pot. That means I need to now identify three catalytic systems. One to accomplish highly selectively this 5-exo dig cyclization. This is not as simple as or as straightforward as earlier cases, because it's sort of complicated with the presence of this olefin, because if I try to activate this alkyne, which is a pi system, this can equally activate this olefin as well, and this amine should not now cyclize onto this olefin. It should preferentially cyclize onto this alkyne. That's what is very much essential for the success of these subsequent steps. Okay, with this hypothesis, we indeed made this particular substrate. You see this? This is a hypothetical substrate, and this is the exact compound that we indeed prepared. We prepared this compound, and we now optimize the system. I'm completely avoiding, throughout my presentation, I'm completely avoiding optimization studies to make it easier and with less distractions. So now this substrate, in the presence of catalytic amount as small as 2 mole% of silver acetate, selectively provides this indoline. Now subsequently optimize the cyclization of this indoline to this indolyl amine in presence of TMS triplet. As you see here, only the electron poor amines work, not the electron rich amines such as benzylamine or PMP amine, para, methoxy, phenylamine, PMP. Okay, so now we can access up to here, but several of our efforts to cyclize or to accomplish this intramolecular hydrogenation were not successful in our hands. Okay, but we didn't want to give up. We didn't want to just give up because we made such a progress. We just had to leave it here is something we didn't accept. We wanted to identify something else, otherwise to actually lead to such systems. In that context, we got an idea. Can we have azide in place of amine? Can we introduce now azide? And this azide, of course, can be envisioned as an amine equivalent if you just employ Schrodinger reaction. Otherwise, a simple reduction of azide. Otherwise, it can also be functionally modified via the 3+2 cycloaddition, leading to such a structure, and subsequent triazole modifications can be done. Probably can lead to tetrahydrobiogabulins. On the other hand, we also intended to actually proceed through a nitrine pathway, and subsequent functional group transformation can lead to tetrahydrobiogabulin. This was the idea. So effectively, to summarize this particular slide, we intended to utilize amine in place of azide in place of amine, but here it required, as you must have noticed, a little different condition. Obviously, that here the acidating agent is now a TMS azide, that is trimethyl silyl azide, and then bismuth chloride as catalyst for this particular purpose. Okay, this is what happened, but we met with a surprise. So what happened in this case when we actually made this compound, and at this stage when we tried a different catalyst to actually accomplish what are the desired azide transformation. What we observed was, in presence of this catalytic amount of palladium acetate, this straightaway provided this beta carbonyl. So if you realize the difference between this structure and what we intended to do, what we intended to do, we intended to access tetrahydrobeta-carbonyl. This is a saturated system. Okay, so but here we obtained completely unsaturated equivalent of it, that is what we call as beta-carbonyl. You can see that the scope is fairly broad, a variety of 1,3 disubstituted systems having arenes as well as heteroarenes, such as this, for example, can be accessed. Not just 1,3 disubstituted, you can even access fully substituted pyridine core, as in this case, for example. You can see that 1, 3, 4 trisubstituted pyridine fused systems also can be accessed in good yields. This was the work that we actually demonstrated here. Okay, now question obviously arises to us at that point of time. Of course, now it is published, it's all clear. I'm writing this structure here nicely, but we're not very sure what was happening during this transformation, during this particular transformation. Are we really accessing this indolyl azide or something else? So for that purpose, we wanted to actually isolate this compound. So what happened, my student, the first author here, Sielmann, so she actually purified the compound by column chromatography and left the sample overnight. And next morning when we came back to the lab, we identified nice big crystals floating in that test tube, in that solvent, in the eluent. So we actually took the crystal structure of it. We identified that that is this particular compound. If you see the difference from earlier structures such as this versus this, is you can see that new hydroxy group got introduced here. Okay, so to expedite this process, I said this happened over a night. So to expedite the particular process, we then just heated up this particular azide. We straight away got this hydroxy incorporated beta-carbonyl. This scope also is quite broad, as you see here. What is impressive about it is there was no method earlier reported to access hydroxy incorporated beta-carbonates. These are very much relevant because several natural products were present with this particular core, this method now actually can be employed to access those natural products as well. Okay, so you can see that variety of 1, 3, 4 tri-substituted systems, that is hydroxy incorporated systems, can also be accessed. Not just that, we didn't leave this work up to here. In addition to these carbolines, we also extended the scope of this work to access other pyridine fused systems, such as, depending on whether it is a arene or a heteroarene, different types of scaffolds can be accessed, such as this pyridine fused benzofurans. You see here these two structures, and pyridine fused benzothiophenes of this kind. It is only representative structures. Again, we made few other compounds. One can refer to the manuscript here listed. Okay, you can even make isoquinolines such as this just by mere design of your precursor. Okay, if you make any of this kind of azido olefins, you can obtain under this condition respective pyridine fused arenes and heteroarenes such as this. Okay, so having told you about hydroxy incorporated method also, so you can also do that by avoiding any catalyst. Just take this compound, heat it up in toluene or any other to that matter, any other hydrocarbonaceous solvent to actually obtain this hydroxy incorporated beta carbolines. You see the scope here. Here also we prepared pyridine fused benzofurans, benzothiophenes, and even isoquinolines. Okay, most important, yes, I demonstrated the scope of this work, but most important is the mechanism. I should convince you how these are forming, obviously. So this, obviously, I already told you that in this particular instance, silver preferentially coordinates or activates, being a pi acid, coordinates preferentially to this pi-rich alkyne, and thus facilitates this 5-exodic process leading to this particular compound. Now this is nothing but a Lewis acid catalyzed a nucleophilic substitution, that is nucleophilic azidation in this particular case, giving this particular compound. Now subsequently is more important to us. That is, now what is this palladium doing? Palladium is believed to decompose this azide to nitrene, which subsequently undergoes appropriately situated olefin insertion, and that subsequent functional group transformation leads to this dihydro-beta carbolin. This is something that is aerobically driven, aerobic aromatization, or otherwise you can also call oxidative aromatization, leads to beta carbolins. This is a mechanism. Okay, I hope I convinced this part, but the other part is more interesting. That is how this hydroxy group is getting introduced. Okay, how this is coming. What we propose is that this is undergoing a Huygens types 3+2 cycloaddition to initially form this compound. Okay, you may question, how do you know this is forming? We have crystal structure. We are fortunate that we could be able to actually isolate crystals of this particular compound. This is the crystal structure of it. And subsequently, under obviously that we are heating this process, this substrate, so this is undergoing denitrogenation of leading to the formation of this dihydro-beta carbolin. Autooxidation gives, and now in this particular instance, the oxidase product subsequent enolization, otherwise you can also call this as aromatization process, gives hydroxy incorporated beta carbolin. Okay, now you may be ready to ask me, how do you know that this particular compound decomposed to give only this one? Why did you place this double bond here? Why can't the double bond be here or here? Probably that should have been more stable, because it's in conjugation with the entire indole system. So for that we have done an experiment, that is this one, where we purposefully introduced an alkyl group at this position. This alkyl group, obviously you can understand that this doesn't let an oxidation of this kind. Obviously ketone cannot form now. This is already a tri-substituted system. So ketone cannot form. We have actually isolated this particular compound. This was very, very useful evidence for us, because not just it proved the hydroxy is coming from aerobic oxygen. It also showed the position of this double bond. As you see here, this double bond is not here or somewhere else, or here, for example. Okay, so thereby we assign this structure analogously to this particular compound. Next, most importantly, how do you know this oxygen is coming from air or water? Water probably traces amount of water may also be introducing or helping this oxygen to enter at this position. So to that we perform the reaction of this compound in presence of the labeled oxygen. Now this is 18 oxygen O18 labeled oxygen that got introduced in the product. The complete characterization of this product revealed that this is indeed happening through aerobic oxygen, not through any other oxygen source. Okay, so that's how we could able to make this particular work culminating at this stage. So, so this also introduced us to, as you see earlier, to the palladium chemistry. You saw that this decomposition occurs via palladium. Earlier I showed you this decomposition of this azide. So while we talk about palladium chemistry, this reaction comes to our mind, which is a textbook chemistry. As you see, this allylic substrate, the Lg refers to the leaving group. Okay, this activated allylic systems in presence of palladium is known to form this pi-allyl palladium species. It's an eta-3 complex. Now this can undergo nucleophilic addition depending on the substitution pattern of this allylic system. It also depends on the nucleophile. It can lead to two types of products. Either this nucleophile can get introduced at this position or this position, leading to these kind of products. This was very well known. This courtesy to the discoveries of Suzy and Tross. This reaction is now named as Suzy-Tross reaction. Fine, up to here we are very much familiar, comfortable. Now what was not very much known is this substrate. Okay, what is the difference between this and this? Now, while this is an allylic substrate, this is a dianylic substrate. It has two double bonds, as you see, in line or in conjugation. Of course, this is also another type of substrate you can think of. Now, what was reported by Tross in early 80s? This kind of compounds under palladium catalysis forms a vinyl pi-allyl palladium species, such as this or this. Right, these kind of compounds can give an equilibrating mixtures of these compounds. Now, introduction of appropriate nucleophile now can give products such as this. Okay, now that means this nucleophile can react here, or if you take this particular intermediate, this nucleophile can enter here or here, leading to these three combinations. This was reported, but we gave it a thought. Okay, can we do something different with this kind of a vinyl pi-allyl palladium species? Okay, while this is a proposed species, why can't this double bond in a ene type fashion react onto this electrophilic species such as this palladium species? Can something happen? Why this was not observed? Can we do something otherwise? So for that, we actually proposed a hypothesis. We came up with a hypothesis. We came up with a proposition. What the proposition says is that if I can introduce across the same dianylic species, if I can introduce substituents such as this R2, R3, for example, okay, they can exhibit a so-called allylic strain. Whenever you substitute two substituents in an allylic position, because of as my hands show, they can experience allylic strain. This is called A13 strain. So because of this strain, analogous to because of this strain, this substrate now can reorganize. It can repel. It can move away from its original position, leading to this particular angle. So that particular angle can lead to these arms, this arm or this arm come together as in this fashion. Okay, if you carefully observe this transition state model, this is the same substrate, that is the vinyl pi-allyl palladium species. I have written in a different way. I have written in a way that it possibly can reorganize and get this double bond in closer proximity to this particular carbon marked in blue. Okay, now question is, can this now undergo ene type cyclization? okay, leading to the formation of the cyclopentene cation, cyclopentenyl cation, sorry. And now subsequent deprotonation can give actually cyclopentadienes of this kind or this kind, depending on how it wants to reorganize. This can also, the overall process can also be called or termed as an Isonazaro type reaction. Okay, so with this proposition, we prepared indeed a substrate having methyl group in place of this R2 and R3. You can see that wherever I listed R2, R3, you see here, this R2, R3 are now methyl groups. Okay, and these methyl groups and palladium catalysis, you see, simple, readily available. Even tomorrow, you can go back to your lab, any of your simple labs. This compound is commercially readily available. Of course, this is a solvent which can be easily accessed. Just take the substrate, treat under the straightforward conditions. Actually, now obtain different type of cyclopentadiene that is not belonging to any of these two which we originally proposed. Okay, this compound can be clearly distinguished from these two by NMR. If you carefully see the structure of 2A, it is a methylene group, unlike these two structures. So that indeed gave us an idea that this is mostly forming another isomer of the cyclopentadiene, giving this particular compound. Indeed, we have a crystal structure, of course, later obtained. You can see that variety of tetra substituted cyclopentadienes can be accessed to this method. Okay, we didn't stop here. Okay, how we can extrapolate this concept? So what we have done then, along this dienylic system, we now introduce a fusion in the form of a arene or a heteroarene. What this fusion does, it can lead to now a fused cyclopentadiene such as this, right? Yeah, so this is acyclic diene, so it gave cyclopentadienes. If I have a fused diene, it can lead to fused cyclopentadienes of this kind. Indeed, our hypothesis worked. You can see that variety of indines can be accessed. These are only representative structures for your information. You can also access heteroarene fused systems such as this benzothiophene fused cyclopentenes also can be accessed. Okay. So this is the scope of this work. We didn't leave here. I'm skipping some work in this, but one may go through this particular paper. But I'll straight away move to an important application of this work that is in the form of this natural product synthesis. So these compounds listed in this box are the ones belonging to resveratrol family of natural products. This is a huge family of natural products that is resveratrols. Okay. So among them, this passiflorol attracted our attention. We thought that this is something that we can access or that we can prepare through our methodology. So for that, we made this appropriate starting compound, this bromoalkene. Then we introduced aldehyde functionality, and then leading to this particular this benzylic alcohol acylation, followed by applying our own method, that is this palladium catalyzed reaction gave us access to this indene. Our intention originally was to hydrogenate followed by benzylic oxidation. Straight away should have given us passiflorol. But what happened? Hydrogenation went fine, but selenium dioxide oxidation, which we thought would give oxidation, actually gave us ore oxidized product. You can see that it introduced even a double bond during these conditions. But nevertheless, when we refer to the literature, this compound was already prepared by Richmond Sarpong for the synthesis of passiflorol. Thereby, actually, this represents a formal synthesis of passiflorol F. So, looking at this strategy, you can also assume that this can be generalized, this strategy, to access other kinds of natural products in this particular family. Okay, this is one application of some simple reaction, textbook chemistry that we see, that is this Suzitos chemistry, or otherwise called as allylic alkylation chemistry, and we extrapolated to a different extent and utilized in the synthesis of these compounds. Okay, now you have seen intermolecular version of it. I hope I am audible to everyone. Everything is fine. It's not like yesterday, I hope.
Yes, it is fine, sir. Continue, no problem.
Yeah. I can move on, right?
Yes, very good voice. Clean.
Thanks. Yeah. So what you have seen from the previous work is the generation of spiral and introduction of a nucleophile that is an intermolecular version. We thought, can you do this intramolecularly in continuation? I'm actually continuing the same Suzitos chemistry. Can you do this intramolecularly? That means now what I have done is I place the nucleophile intramolecularly. rather than bringing it from outside, right? This nucleophile, now when this spiral-LPD generates, this nucleophile probably react in a 5 endotric type cyclization, can probably give me access to cyclopentenes of this kind. Okay, so in this context we need to learn few things. That is, in place of, based on our experience, this leaving group is considered to be this acetate. This is based on the earlier work just now you saw. Okay, this is the design. Actually, this is the primary design. This is literally written on the piece of paper, and we wanted to now start our initial discussion. So what we thought, okay, fine, up to here there's no issue, we can generate PyLLPD. Now this double bond, being part of an arene or a heteroarene or an olefin from outside, can react in 2 modes. One is a 3-exotrig cyclization, okay, which is actually Baldwin, according to Baldwin, it is favored 3-exotrig cyclization. This can also undergo in the path B, as shown in path B in red. Okay, this double bond can offer in-type cyclization onto this center, which is connected to R1, R2. That actually represents a 5-endotric type cyclization. This is disfavored by Baldwin. Please note this point. Okay, this is a favored cyclization, this is a unfavored cyclization, but otherwise, if at all we can accomplish this cyclization, this can lead to obviously a quaternary or a spirocarbon, depending on the style of these R1 and R2 are placed. It can also lead to cyclopentene fused arenes and heteroarenes, depending on what kind of design you are coming forward. Okay, this is a hypothesis and background. Okay, now based on this hypothesis, we designed this substrate. As you see, this is a hypothesis, this is a hypothetical substrate, and now we optimize. Again, I'm skipping the complete optimization studies, just mere employment of palladium chloride in toluene. Now you can access structures of this kind. What is attractive compared to the earlier work is that now it introduces a quaternary carbon, right? It introduces, you can see from these examples. Okay, we didn't we generated complex tricycles, tetracycles, okay, even pentacycles were also reported, such as this. Okay, as I categorized on this particular slide, 2 important features of these examples. One is all of them incorporate a quaternary carbon. You can see here, 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 and here. And this red bond, obviously, by now you must have understood, is a new bond forming. Okay, this is a new bond forming here between here and here. This is a new bond. Okay, now this represent indines. Okay, we made a variety of fused heteroarenes also, such as these benzothiophene. Okay, few other examples can be found from the paper. Otherwise, we also made fused indoles as well. You see this kind of complex natural product like structures. You can see these pentacycles such as this. Okay, they all incorporate, as you see, quaternary carbons. Okay, so this, some of these compounds, a very interesting part of these compounds is showing very much promising biological activities. Okay, the details are still under consideration, so I'm not revealing the details of those biological studies, but these are under process. Okay, through this slide, I have demonstrated the creation of a quaternary carbon. Okay, quaternary carbon represents, as you all understand, by definition, by IUPAC, a quaternary carbon is the one whenever a carbon connected to all 4 carbons, definitely has to be all 4 carbons. Many people use that in different contexts. Not that carbon connected to any other heteroatom is a quaternary carbon. It has to be connected to all 4 carbons. So in this particular instance, you see that we indeed have generated all carbon connected quaternary center. Okay, then we also extended the scope of this work to the generation of a spiro center. What is a spirocarbon? This is also a quaternary carbon, but by virtue of this substitution, you can see that this can be now called as a spirocarbon. We prepared this substrate. This is oxindole based substrate, almost under the same conditions, catalytic amount of palladium chloride in presence of dichloroethane provided access to, you see that variety of spiroxindole. The arrow necessarily represents the difference in these structures. You see that benzothiophene here, the sulfur facing the top, here the sulfur facing the down. That tells you that depending on the type of starting compound you make, you can make whether this kind of substrates or the other kind of substrates as shown here. Okay, this represents benzothiophene fused spirocyclopentene oxindoles. Okay, and we also demonstrated this in the synthesis of spirocyclopentene fused indoles as well. You can see that you can make other type of indoles. This is one type. All these 4 represent one type. This is another type. There are few natural products in this particular core also. We actually attempted a couple of natural products. Somehow we couldn't finally synthesize, but I'll show you in the coming slide the synthesis of other natural products. We can also prepare indene, Spiro indene fused oxindoles as well. Okay, what is impressive or interesting about this particular strategy is this particular one. Okay, so whenever this R1 is unsubstituted, okay, this reaction doesn't work, mind you. Okay, this reaction doesn't work. So you can see that whenever this R1 is a hydrogen, this reaction only gives deacylated, deoxygenated product otherwise. Okay, this is of course, this kind of reaction is very well known with palladium chemistry. Okay, this is just underwent protonation. Okay, so but otherwise we also demonstrated the substituent role also through this method. Okay, so now I'll show you how we applied this in the synthesis of natural products. Okay, these are the natural products called thiabinaquinone and dichroinone. Okay, these are simple compounds. What is impressive about it is the presence of this particular quaternary carbon and interesting benzene benzene type structure here, fully substituted or fully functionalized, I should say. Okay, in the form of a quinone. Okay, how we have accomplished this? We started with this particular trimethoxy bromo derivative. Of course, it was prepared following the literature protocols. Now this was elaborated to this acetate following a simple chemistry. Okay, you can of course see this publication. This was published on this year very recently, and then applying our own method, that palladium catalyzed 5 endo-trig type cyclization gave us access to this tricyclic core oxidation, that is this CAN mediated oxidation gave this quinone, demethylation gave us access to dichroinone. Okay, so mechanism, which is more important, how what might be happening here. Of course, by now we must have understood the mechanistic features of this particular compound. First step obviously is the formation of the pi-allyl-PD. You can see that it forms a pi-allyl eta 3 complex of palladium. Okay, palladium 2 is believed to undergo reduction in situ to palladium 0 in an analogous to Wacker type process, and this palladium 0 is now responsible in forming this pi-allyl-PD. I told you, if you remember, the role of substituent. This particular role is envisioned in the form of this steric hindrance, leading to the reorganization of the substrate into this form. This is effectively, if you carefully see this particular form, is actually bringing this aryl system and this particular reactive site, that is this reactive site, closer compared to this. You can see this arene is away from this reactive site in this particular mode, but by virtue of this substituent, we believe that this actually bringing them together, and subsequently you can of course write the same substrate in a same intermediate in this form, that now undergoes N-type cyclization, giving access to this cyclopentane pentane complexed to palladium, and decomplexation and deprotonation gives access to these particular compounds. Okay, this is a mechanism. I can tell you at this point, there are other types of mechanisms possible, though I am not going to confuse you with more details about this particular reaction at this point. These are all under computational investigation at this point. We are also working on achieving experimental evidence in the form of NMR studies, and of course we have already performed. I actually skipped the details, kinetic isotope effects, as part of this particular study, to show that this particular cyclization is not going through CH activation. This is actually going through N-type process. So those details I skipped. Actually, if someone interested can go through this particular paper, or one may contact me. I can actually share that particular paper. Okay, one more last concept. I'm going to wind up my talk in just 3 slides by introducing one more concept within this palladium in chemistry. Okay, so having seen these kind of propositions, like in the earlier case where we generated pi-allyl palladiums and a double bond appropriately situated reacting onto the pi-allyl-PD in an N-type manner, we thought, can we have a design such as this, where the double bond is actually an allylic system from this position. This is a skipped diene, unlike earlier was a diene, and this is a skipped diene. right? Now, what we thought, if you can follow the hypothesis, subsequent work will be easy to follow. What we thought was, under the influence of palladium, just like the earlier case, can form PyLLPD species across this system. Now, this double bond can offer ene-type cyclization, giving this cationic species. Now, deprotonation can lead to mixture of regioisomeric mixture of these compounds, but eventual oxidation probably can give us access to the benzenoids. Why I called benzenoids? If this arene is a benzene, it is an naphthalene. If this is a heteroarene, this is again depending on whatever that heteroarene is. For example, in this case, if your heteroarene is a indole, you can call this as a carbazole. Right? You can see the representative structures employing this particular hypothesis. We demonstrated the synthesis of 1,3-disubstituted carbazole. These are again representative structures, and good yields were obtained. And this is again, you can see the straightforward nature of this reaction. You can also execute this chemistry in your lab, just provided you make the substrate. You can execute with no great significant cost implications. Okay, so what we observed during this process, or you might have also observed, this double bond is substituted here. Okay, please let me take your attention to this. This double bond is substituted here. Okay, if the double bond is substituted, this is what the product we obtain. Okay, when this double bond is not substituted, that means when this R group is H. Okay, when it is a simple allyl group here, we observe a different product. Okay, what we observed is the formation of the cyclopentene fused arenes and heteroarenes. Okay, what is happening here? What is happening was that, of course, this proposition came out of experiments. Especially isolation of this particular compound was very important to came up to this hypothesis. What we believe is that first this undergoes double bond isomerization. This allyl undergoes isomerization to this vinyl system. Now this undergoes metalloene reaction, otherwise palladium-ene reaction, however you call. Metalloene is because we are using a metal, and metal is inducing the ene reaction, so we call this as a metalloene reaction. We can also call palladium-ene reaction. So that leads to this formation of this cationic system. Subsequent deprotonation, isomerization gives fused cyclopentenes of this kind. You can see the scope of this particular process. Okay, I'll explain to you mechanism in detail, but you can see that again we have developed yet another process for the formation of cyclopentene fused heteroarenes. Okay, what is more interesting here is, instead of allyl, when we use butynyl group to see what can happen. Actually, our intention was to see whether whether this butynyl can undergo cyclization, can this lead to higher member rings, but it didn't happen. It was actually undergoing, very surprisingly, as you see, double bond isomerization. Okay, it's a twofold olefin isomerization giving this, and subsequent cyclization giving us the same product as you see earlier. Okay, that means whether you use allyl or butynyl group, you probably will get a similar product. Of course, the side chain is different by virtue of being unsubstituted. This is a methyl group. Because of this being butynyl group, this is a ethyl group here. Okay, so that's the work I thought of talking to you. This I'll wind up with this mechanism. Okay, this is how the side product forms. This is nothing but a reductive elimination process gives this side product after the oxidative addition. Okay, so this was even evidenced by the deuterium experiment. When we performed this reaction in presence of D2O, we saw that this product was actually getting deuterium at this position. That tells you that perhaps that in situ is forming a palladium hydride species. Perhaps I use the word, and that deuterium or hydride is getting transferred in the form of this D to this particular benzylic position. Okay, you can see this is considerable. This is 50%. So that tells us that this is coming from the moisture present in the reaction medium. That means, so sometimes many people think, or many people focus on the actual product. You can see from the last two slides how isolation of impurity matters, how isolation of an impurity matters. Okay, so this mechanism actually heavily dependent on the isolation of this particular compound. If the student would have thought that this is actually okay, 10% only, doesn't matter, we would not have got this kind of mechanistic insights about this particular transformation. So through this particular slide, and at this juncture I want to suggest all of the organic chemistry practitioners, please focus on impurities as well. I can tell you that can give you several of our works, probably you would have seen, they all came out of serendipity. Most of them are not planned. Though I showed you, I projected as if they are planned, but majority of the works, I've talked to you probably 2-3 works here, or 3-4 works here. But if you refer to our other works from other fields of research, most of the works are unplanned. I can tell you this serendipity favors those who actually pay attention to small details. I can tell you, with my experience at least. Okay, so okay, now explaining the formation of the actual product that formed. This is impurity that formed. I explained so far. The actual product forms via metalloimine reaction or palladium reaction. This cation now undergoes elimination either from the alpha position. If it undergoes proton elimination, undergoes from this position, it forms this one. If it undergoes proton elimination from this position, that is gamma prime position, it forms this one. These two isomerize eventually via the processes mentioned here, leading to a single compound, that is this. Okay, so that's how we can explain the formation of these products. Okay, with this I summarize that I talked to you about two concepts. In concept one, actually I didn't show you complete works because of the paucity of time. Dr. Jagan gave me half an hour, 40 minutes, so I should ensure that I don't take much. I hope I'm on time, but otherwise this is what I've shown. Very briefly, I talked to how this kind of substrates, these amino inols, can be extended to cyclopentene fused indoles such as this. And I also showed you how beta carbolines were synthesized starting from similar designs through this particular work. This is something I deleted in the last minute, thinking that I may be exceeding my time. So, but otherwise you can also make compounds of this kind. This work is something I've excluded from today's talk. In concept 2, which is based on allylic calculation reactions or Suzy-Trost type chemistry. So I showed you how we can simple chemistry can be extended to complicated structures such as this through this particular work. And very recently we published a generation of quaternary carbon and Spiro carbons as well through this particular work. And I also showed you at the end how carbozoles and cyclopentene fused deuteriums can be generated. This was the last work I talked to you. Okay, for more details, please, I would suggest all of you to go through our web page. We, as I said earlier, we work on metal-free chemistry also, especially employing phosphines as catalysts. That's a huge area we developed, but I didn't talk about it at all today. With this, let me thank my research group. So far, 4 graduated from my lab. These are the other students currently pursuing research at different stages. This is a somewhat old picture. A couple of people. This was a postdoc working with me. He recently completed his tenure and left. And he also left for PhD elsewhere. He was a BSMS student. Otherwise, funding came. This is very, very important. Funding came from Aysar Mohali. Aysar Mohali is generous enough to support us or the period. And then, of course, DST for the generous funding through this particular scheme. SERB for the fellowship. Okay, you can of course contact me through these emails. You can also refer to these numbers are a bit peculiar because there were some domain issues, so our computer center couldn't resolve this issue so far. But you can remember these numbers, and this is our website. Otherwise, you can reach our website through Aysar Mohali webpage as well. Okay, thank you very much, Dr. Jagan. Thank you very much, Dr. Srinivas and others for the introduction, and thank you all the participants for the kind attention. Thanks a lot.
Thank you, Dr. SSV Ramashastri garu, for your nice presentation.
Yeah, is there any questions?
Yeah, now it is open for questions. Any questions you have, please interact with the speaker. Now I request all participants, if you have any question, raise your questions. Okay, I will start with my question.
Yeah.
So you have explained in our presentation that palladium chloride, PDCl2, which you used for the synthesis of cyclopentadiene from a dialkene.
Yeah.
So 10 mole % you have used that.
Yes.
PDCl2. You said that a very good reaction you can do at laboratories, common laboratories, wherever you have, you said. So this all not only palladium chloride, and we can try with some more other catalyst also the same
Yeah, but
reaction.
the best yield was observed with palladium chloride. And
Okay.
as you mentioned, 10 mole percent is something we have shown for publication sake, but you can even go down to 5 mole percent or even less. Only concern in that case will be reaction takes more time. Product
Okay.
you can obtain in almost similar efficiency, but reaction takes more time. So as I said, 10 mole percent is fine from academic point of view. Wants to pursue this further, they can go down also, and we have indeed this up to multigram scale as well. So we found it's an efficient reaction. It is executable in higher scales as well.
Very good. Thank you. And my other question is the same reactions and similar kind of reactions can we allot to, especially to our MSc students, those who are pursuing their project work some 2 to 3 months. In that aspect, can we purchase these starting material dialkenes and catalyst, or we have to prepare
These are commercially available. These
available?
catalysts are commercially, yes, commercially available, cheap. I can tell you. I think 5-10 gram will cost 1-2 thousands or something. To my
So,
knowledge, to my knowledge.
okay.
Okay, so I don't remember now, but this is yeah, this is quite cheaper. Even one can use palladium acetate kind of catalyst also, which is to my knowledge even more cheaper. Palladium chloride is something very cheap. So one can definitely do it. It's all about catalyst wise. I don't see any issues, but it's all about what kind of starting compound you are preparing, how you are preparing. Probably the starting compound synthesis may involve some cost because you need to identify right or appropriate starting compounds, and it takes about 3-4 steps. So the particular student, I'm sure a master student should be able to handle. I didn't talk about all those details. In fact, I didn't in fact many of our of the authors are master students actually, so they can happily do this chemistry, and this should be very much fine.
Thank you. Definitely, I will contact you regarding this to plan a small project, especially for the
Definitely,
benefit of students.
definitely, yes.
So that will be more useful. Whatever you said, lighting chemistry is very good because we have on the syllabus also that
Yeah,
CC bond formation,
yeah, right, right,
especially you know that
right,
in the third semester, especially we have that CC bond formation by using palladium catalyst. So
right.
palladium chemistry is there. So many Suzuki
Yeah, this is
coupling
also CC
reactions.
bond forming, but it's not through traditional chemistry
Yes.
such as
Yes.
Suzuki or Negishi type coupling. This is through Suzuki Toss type chemistry.
Yes,
And
actually,
this also involves
for the participants, they need whoever the participants in this webinar, they need this practical approach and research how the reactions will go. But theoretical aspect is different and practical thing is different. You know
right,
very
right,
well.
right.
That's why the participant they are eagerly seeing about the practicality and the research reactions, how it works in the laboratory, especially as you said that yield is very important, nah? So
Right.
that's why
These
these
are
kind of things are very
reasonably high
important.
yielding. So I don't believe in 99% yields, because whenever I see in publications also, I usually don't believe 99.
Yes.
Okay, so I'm very happy to report 70%. No problem for me. So, but concept is something that carries a publication, not 99% yields. Yeah, but otherwise, one more thing I wanted to inform not only to you, to all the attendees also, is obviously that these papers come up with supporting information. So in that, detailed procedures, data, all the spectral data also described. So one may kindly go through that. Otherwise, if someone is not able to access, one may contact me also. So through my web page, all the details are available. Either Mohali web page or my own web page.
Thank you, Ramashastri garu. Any questions from the participants? Okay, you can ask. Any more questions? Please. Triveni is there. Dr. P. Triveni, you want to ask any question?
I think Dr. Rajashekar Reddy is, I think,
He's asking question.
raised the hand here. I
Okay.
think.
Dr. Rajashekar Reddy, you are there? Are you there? Please ask your question. Please raise your question, Rajashekar Reddy.
I think he has to unmute. He has to unmute.
You unmute, please.
Yes, yes. Are you able to hear me now?
Yes,
Yeah,
yes.
yes,
Yeah,
yes.
Dr.
Yeah. Sir, it's excellent lecture, sir. This Dr. Rashek Reddy, hope you know me. I'm
Yeah.
your immediate junior to you at Ideal College, Kakinada.
Okay, okay, nice.
Hope you remember me. Sir,
Uh,
I have just, it's an excellent work and then wonderful work, and it's a great hard work, I feel. And
thank
it's very systematically organized and very well informative and well discussed and good presentation, sir. So I just have slide number 8. I have some small queries. Slide number 8.
you. Yeah,
And
can you see now? Slide 8?
yes, yes, I could see now.
Oh,
Most of these reactions are called out in silver or arm chloride like this when there is alkyne and there is nucleophile. And why people are unable to do these reactions with simple copper chloride, or have you attempted whether this reaction has any scope to do it using the copper or other simple metals like zinc chloride or copper chloride and other?
right.
Yes.
Yeah, that's a very, very valid question.
Hmm.
Is your question complete or you're still
Yeah,
asking?
still I'll ask next after
Sure.
this.
Can I
Yes,
ask a question?
yes, yes, please.
Yeah. So, as you rightly pointed out, we have done this kind of cyclization with gold and silver.
Yes.
You also would have caught my attention when I was saying these are pi acids.
Okay. Hmm.
The pi acids. Pi acids mean there are two types of acids within Lewis acid. Okay.
Okay. Hmm.
Broadly, acids can be classified into Lewis acid, bonded acids, but within Lewis acid, there are two types of again acids. One is regular Lewis acids, which go through sigma bond formation.
Okay.
Otherwise, there are some acids which go through pi activation selectively.
Okay. Hmm.
So this activity, why these particular metals, for example, gold, silver, lead, mercury, these kind of metals, they selectively activate pi system. There is a theory behind that. I will skip that portion at this point. But
Okay.
different types of metals have different types of properties. I cannot use, for example, zinc chloride, magnesium chloride, because they behave like a traditional Lewis acids. I mean, they may potentially activate my OH, which is unintended at this stage. So
Okay.
I want a Lewis acid which needs to be activating the pi system in the form of that alkyne, especially among even alkene and alkyne. So I need to achieve the specificity among the three types of functionalities present in the starting compound, if you see carefully. Right? So there is a OH, there is alkene, there is alkyne, there is amine also. So a metal should not get bound to either OH or amine and get deactivated. And the metal should not also activate alkene, and I want preferentially activating pi-rich alkyne. So for that purpose, it is very well demonstrated even in the literature also that gold, silver kind of salts preferentially activate alkynes, and once it is activating alkyne, as I said, pi activator or pi acid, these salts now induce more electrophilicity into that particular bond and subsequently facilitate the nucleophilic addition of these amines.
Yes, I agree with you, but we also tried with this where there is a amine and then alkyne is present with instead of earlier it was reported with the tandem cyclation reaction. I hope one of my paper we have tried with alkyne and amine containing compound. We did some cyclation reaction coupled with amino alcohols and all. But have you tried with copper chloride? Is this a transformation? Have you
Actually,
attempted?
I forgot to address that. Yes. So, as I said, different metals have different properties, different ways of activating these pi systems. Copper also activates. You are very much correct. Okay, copper also is pi acid. Okay.
Yes, yes.
But here, if you see carefully, it underwent, the amine underwent 5 exodic cyclization. Copper gives 6 endodic cyclization.
Okay, six membered cyclic. Okay.
Yeah, so that is a difference. We don't want that 6 endo.
Okay, fine. So one more question, sir. Do you notice any other slide number 9?
Okay. Right.
Do you notice any other regioisomer here? Here at Popa, here you are getting the you have alkyne system here, and 6-membered and 5-membered ring. After coupling this one and here, is there any regioisomers here? We got any
No, no,
selective?
you didn't observe. This proceeds to high selectivity. I cannot say specificity because I'm not to that level confident as of the analytical tools that we use, but
Okay.
it goes to high selectivity.
Okay.
Yeah. We didn't observe other isomers. That's for
Other
sure.
isomer. Yes, sir. Okay. Okay.
Yeah.
So maybe the reason, any reasons are for that? Any reason that
Again,
you are collectively
points to
getting?
the same that whatever silver I'm using here, it must be highly selectively binding to the alkyne. Okay. And that is now activated in preference to the other functionalities present in the molecule, and that must be leading to the product that we are seeing.
Okay, okay. Okay, thank you, sir. Another one more doubt. And slide number 14, sir. Now is query.
No problem. Slide number 14.
Yes.
Yes. Okay. Which state? These are several animations here.
Yes, cell animation, sir. Can you just give a full animation? You have done some 18 labeled oxygen, because actually I'm not, we don't have that much facilities here to do this. But I just want curious to know that how do you get a source of this 18 oxygen? And once the reaction is done, how do you characterize them?
Yeah, no, characterization is not a big problem, but getting that labeled oxygen is the biggest problem.
Yeah, yes.
So I was fortunate. Actually, my colleague at that time had this labeled oxygen cylinder.
Okay.
100 ml. For example, 100 ml costs multiple thousands. It's very costly. Okay. And getting this nowadays, I don't think Sigma. That was a Sigma Aldrich product. Nowadays, I don't think Sigma Aldrich is supplying this labeled oxygen at least, because I used a cylinder from my colleague, but no, this was done almost 4-5 years ago. Till now, I couldn't replace to him. Though I have money, I couldn't replace because the product is not available. And I think that's the problem. But otherwise, characterization, you can characterize happily by mass.
Okay, mass only. Okay.
Yeah, you will see M+2 peak as.
Yeah, M+2 peak. Okay. I got it. So I thought, is there any NMR techniques can influence the chemical shift and all that? Okay.
NMR cannot differentiate by mass, because if it is simple oxygen, that is O16, right? Regular oxygen, O16. This is oxygen 18. So we should see M+2 as a base peak or major peak.
Okay. Good, sir. I understand that. And one more query, sir. Slide number 25.
Okay.
Sir, Jagan sir, do you have time, or shall I already posted that?
Okay.
Yes.
Carry on.
Okay, only one question.
Okay,
sir.
okay, carry on.
Here, what would be the product without protection of TS on indole NH?
Oh, the reaction doesn't work actually.
Doesn't work. What about with the N-methyl group?
Protection has a role.
Any electron donating group such as N-methyl?
Electron donating, it doesn't work.
Doesn't work. Only you required a strong electron withdrawing TS like.
I think electronically that is well placed with the electron withdrawing group such as tosyl or BOC. But BOC is a very labile group, so tosyl being little rigid. So we continued all the studies. You can see across my presentation all the way. Usually protected with tosyl. It's usually rigid. It can bear unless you use extreme conditions.
Okay, it requires only the withdrawing affecting group, right?
You're right. Withdrawing nitrogen it requires.
Okay. So what about this cyclation with a simple, instead of this palladium, with other trans metal free like iodine and all? Have you tested, sir? By any chance?
Iodine, it may work, but that's not what we wanted to develop.
Okay.
Yes, I understand. So see, these results, though I projected throughout my presentation as if I planned, I written on the piece of paper, I told you. Seriously, not all of them are planned. Many, for example, this particular result, which we are on slide 25, is completely unplanned. It came out of serendipity. It came out from nowhere. So reaction as part of, as you know, when you develop a methodology, you explore substrate scope, different things. So during that, we identified.
Thank you, sir. Okay, sir. Thank you, sir. I think.
Yeah. If you have any more questions, please. You can.
I'll post you anyway. You are well known to me.
So you can contact Shastri Garu.
Yes.
Because he is your senior, and you contact him directly.
I know him well, so no problem. I can mail him.
Yeah, Ramashastri garu, Rajశేఖర్ Reddy is my student when I finished my MSc. First,
Oh, great.
I
So
taught him.
I don't know. I know that he was my.
He's working as an associate professor at Vellore Institute of Technology now at
Right,
Vellore.
right. Yeah, yeah, right. I know him.
Yeah,
We were in touch before.
yeah, yeah.
Yeah, yes.
So, okay, Rajశేఖర్ Reddy, you contact Dr.
Yeah, yeah.
SSV
Sure,
Ramashastri.
sir, sir. Thank you, sir. Thank you, sir. Thank you, Shastri Garu. You are
Thank
a
you.
wonderful lecturer and excellent. You are doing a great work, really. So I think it requires a lot of hard work and dedicated students. I think you have that.
Of course. Yeah, yeah. Right,
Um,
right.
And
Thank you
Thank
very
you.
much.
one more point, Shastri garu, one more point. You said in our presentation, there is a one. You said that before, after this palladium chloride cyclopentadiene, I think before this or after, I don't remember the exact slide number. You
Okay.
said that you in your lecture you said that remember this point that favored
Hello.
reaction, disfavored reaction like that, right?
Ah,
Favored or
okay,
disfavored.
okay.
Yeah.
Yeah. So that is here. Yeah, here. Yeah. So
Yeah, yeah.
here
You
is
you
the
you, you have mentioned very right
is
important point that
it's
one
a,
is favored, another one is disfavored, right?
yeah, but we got this product.
Oh, yeah. Okay.
So that
Okay.
also raised a question because we got a product that was disfavored by Baldwin rules. We
Okay.
also pursuing this computationally as well. Why this went against Baldwin rule? Or can we really call this as piezotrick, endotrig, or is there some other mechanism operating? These are all under investigation now.
Very interesting and
Yeah,
very good
right,
chemistry. Very good chemistry.
right. Thank you.
You have mentioned that. Thank you, Dr. SSV
Right.
Ramashastri garu, for
Thank you.
spending your valuable time.
No,
Even
no.
though you are busy with your academic work and academic schedule and research work also. So, from all on behalf of our Adhikavinana University and on behalf of our participants, so I'm expressing my gratitude for your spending your valuable time.
Thank you very much.
All our participants will be in touch with you with their
Definitely.
queries or whatever the questions they have.
Yeah.
Thank you, sir. Thank you.
Thank you.
Have a good day. Thank you.
You too.
Thank you,
Thank
sir.
you.
Now we have other lecture with that. Sir, Jaitirthara sir, you were there?
నమస్కారం అండి.
Sir, namaste, sir. Bonara, sir.
కెన్ యూ హియర్ సార్?
Yes, sir. Yes, sir. How are you, sir? How are you, sir?
ఓ గుడ్, గ్రేట్.
So now we have other lecture with Professor V. Jaitirtharao garu. He is a emeritus scientist from CSIR, Council of Scientific and Industrial Research, IICT, Indian Institute of Chemical Technology. He is working in the field of organic synthesis, and he is going to deliver on this pharmaceutically favored heterocycles via Bailey-Sielmann chemistry, which is a more famous reaction, Bailey-Sielmann reaction. Now, before that, I request my colleague, Mr. Vidyasagar, to introduce our speaker to the participants. Now, I request Vidyasagar to introduce our speaker, Dr. V. Jaitirtharao garu. Please.
Thank you. As I am audible, sir.
Yeah. Yes, yes,
Uh,
you are audible.
Good afternoon to all. This is PSR Vidyasagar from Department of Chemistry. It is my great pleasure and honor to introduce today's speaker, Professor V. Jaitirtharao garu, to this FDP program. Professor V. Jaitirtharao has obtained his PhD under the guidance of Professor V. Ramurthy in 1983 from Department of Organic Chemistry, Indian Institute of Science, Bengaluru, and postdoctorate under the guidance of Professor R.S.H. Lu, 1983 and 1984 at the University of Hawaii, Honolulu, USA, and research associate under the guidance of Professor Koji Nakanishi, 1985-1986 at Columbia University, New York, USA. He was head deputy director and chief scientist of Fluoro Agrochemical Division of Indian Institute of Chemical Technology, Hyderabad, which is a premier R&D institute in India. Presently, he is emeritus scientist at Fluoro Agrochemical Division, Organic Chemistry Division 2, CSR IICT Hyderabad. He is Alexander von Humboldt Fellow, Germany. He visited Germany as panelist for Euro India meeting. He was visiting scientist at Tulane University, New Orleans, USA, and also at University of Miami, Coral Gables, Florida, USA. He is honored with Fellow of the Royal Society of Chemistry London in 2008. He is a fellow of Andhra Pradesh Academy of Sciences, Hyderabad. He is a fellow of the Indian Council of Chemists, Agra. He is the fellow of Indian Chemical Society, Kolkata. He is a member of National Academy of Science, Allahabad. Editorial board member, Indian Journal of Chemistry. Editorial board member of Archivoc Journal and Tribunal Board. Uh, uh, Chartered Chemist, Royal Society of Chemistry, 2016. Founder Fellow of Telangana Academy of Sciences. Recipient of Gadwal District Distinguished Scientist Award. Platinum Jubilee Honor from IICT. Ambassador University, Germany. And his areas of research of interest is organic materials for application in solar cells, light emitting devices, optical sensors, synthesis of heterocyclics for bioevaluation, organic photochemistry, method development for APIs, analytical studies, process development and technology, pesticides and intermediates. So he has published greater than 230 publications and greater than 80 patents. Several are under process. 7 book chapters, 2 books and 2 bulletins, 1 review and 4 technologies demonstrated. He has trained approximately about 44 PhDs and greater than 160 master students for his credit. And presently there are 7 PhD students are working under his guidance. So with this, this is brief introduction about Dr. V. Jaitendra Rao garu. So thanks to Dr. B. Jagannath Reddy sir for giving me this opportunity. Thank you, sir.
థ్యాంక్ యూ అండి విద్యాసాగర్ గారు మీరు కొద్దిగా హెల్ప్ చేయాలి మాకు నేను నెక్స్ట్ స్లైడ్ అంటే మీరు చెప్పాలి ఓకేనా
విద్యాసాగర్ గారు చేస్తారు సార్ దిస్ ఇస్ జగన్మోహన్ రెడ్డి సార్ సో
ఈజ్ ఇట్ ఆడిబుల్
yes sir is it
okay good very good
so you
let me
just tell with Vidyasagar, he will help you sir. Thank you, proceed sir, proceed please.
Okay, hello. Hello. హలో విద్యాసాగర్ గారు
ఎస్ సార్ చెప్పండి
వినిపిస్తుంది కదా
ఓకే సార్ ఓకే సార్
ఓకే let me thank the authorities of Adikavi Nannaya University Rajamendravram Vice Chancellor garu and then in particular thank Jaganmohan Reddy garu convener head of the Department of Chemistry for making me to attend this webinar. And of course, thanks to the Vidyasagar garu for your kind introduction. I need your help also further to take care. So what I would like to say first is to andari sadasulaku shubhodayam madhyanam ayindi aina gani shubhodayame. What I will be discussing with you in about 40 minutes 35-40 minutes is about the pharmaceutically favored heterocycles via Bailey-Sielmann chemistry. So we got involved in this Bailey-Sielmann chemistry for the past 14-15 years, and we have several publications and also a review on this. I thought I should bring it to you because this falls with the medicinal chemistry as it mentioned in your brochure. And the great thing about this, whatever our research is, very simple molecules, very low cost chemicals we have employed, and then using the Bailey-Sielmann chemistry, converted them into the heterocycles, and then taken further for activity, biological activity, and then identified the hits, and then gone to the identifying the leads, and then we got into the animal model also. So further, we are getting into the further studies, whatever possible on that. A couple of examples we have it, we mentioned in that. And then we thank the CSIR, of course, IICT, and then Mera Bharat Mahan, Alexander von Humboldt Foundation, RSC London. The next slide, he is actually the the bold person in this photo is Georges Hammond. And he visited us when I was doing PhD at Indian Institute of Science, Bangalore. That is the way back to 1979. What he says is, many chemists make chemicals but don't evaluate the properties. That is the problem we have it in India. Over 99.9999 scientists will make novel compounds and all kinds of things, and then but they don't look into the properties. This point was raised by Dr. Pavan Tagapur also. You might have heard about him. He's very, very, interested in talking to people to go for patents rather than publications. That's what I understood from him. So even this old man also says same thing. In this photograph, the last person is me. That is during 1979, 80. The third slide, this is about the research interest, as already mentioned by Vidya Sagar. So I just skip it. Then we go to the concept. This is the concept we initiated to look into the various organic materials and then fluorescence properties, biology, interfacing. It's an interdisciplinary research which we initiated based on simple concept that the highly polarized charge transfer state. I'm not discussing about it. The next slide, this is about the structure based and scaffold based new chemical entities which we prepared using Baylis-Hillman reaction. So what we did is we prepared Baylis-Hillman products, converted them into all kinds of heterocycles as shown in this slide, and then we are taken for bioevaluation. Couple of things we put forwarded to the level of animal studies also. Recently, that is the 2018, we have published a review on the work we have done, and particularly the the Baylis-Hillman adducts and their role in modern drug discovery. This is 2018. That's almost 2 years back, which we have published. Okay. So here onwards, you will have more chemistry. That is, we initiated this for making this pyridine carbaldehydes. So tri-substituted pyridine carbaldehydes, and then tetra-substituted pyridine carbaldehyde. You can see plenty of carbaldehyde which we have prepared are shown here. Okay. So how do we make it? So what we prepared, enamide, you can see it here. An aldehyde reacts with amine, gives you Schiff base, and then you treat the Schiff base, acetylate it, that is treating that with the acetyl chloride or acetic anhydride, you get the enamide in abundance. Okay. So if you look at into the the starting materials, aldehyde and amine, they are cheap, available, readily available. And then Schiff base, aldimine, you can make it, and then use acetyl chloride, which is also very cheap chemical. You can convert that into enamide. Like that, you can see it, variety of enamides which we have prepared using this chemistry. Indeed, we have used the amino acids also, where the chiral center gets flat. Okay, no problem. But we converted that into an enamide and used it for converting that into a pyridine carbaldehyde. So whatever the enamides which we have synthesized, converted into pyridine carbaldehyde using Wills-Meyer-Hack reaction. So what we did here, as Pavan Tagapur mentioned to you, that we patented it also. US patent, European patent, Indian patent, all patents we have, this invention. And also we published it in Tetrahedron 2006. So these are the whole bunch of, pyridine carbaldehyde, tetra-substituted, tri-substituted pyridine carbaldehyde prepared. So the reagent what we use, POCl3 or diphosgene or triphosgene. So when you use the POCl3 and DMF, so we do get the product, but it is in low yield. The problem is the phosphorus salts which remains in that round bottom flask of the reaction mixture is the troublesome. So when we change it to diphosgene or a triphosgene, okay, the reaction was smooth. It will be more greener and cleaner because you don't get any inorganic salts here. The diphosgene and triphosgene will get converted into carbon dioxide and left to the environment. So the DMF is the, reacts with diphosgene and triphosgene, gives you the Wills-Meyer-Hack, the reagent that I will show you the next slide. So this, we patented it. The invention or the inventing point is use of diphosgene and triphosgene to get higher yields over 96%, 95 to 96% yield of carbaldehydes, what we have produced, that we patented it. So this slide, it shows the mechanism of this chloronicotinaldehyde formation. POCl3 reacts with, I'm sure you might be knowing it. So you get this Wells-Mayer reagent. Okay, this is an equilibrium, and that reacts with your, with this enamide what we have prepared. The first thing we get the chloroamine, and then again another Wells-Mayer reagent reacts with this. You get again monoformylated bisenamine, you get it, and then this did not undergo cyclization as per our observation, but this can undergo another, Wells-Mayer hack reagent formulation to give you diformylated bisenamine, or this bisenamine can react again with another Wells-Mayer reagent to give you monoformylated bisenamine, and this can convert to diformylated bisenamine. And then further, you can see a lot of arrows is being, shown on this slide. Okay, so that will get into a cyclization and then elimination of dimethylamino group and then hydrolysis, and together all you get the finally a trisubstituted or tetrasubstituted pyridine carbaldehyde, 2-chloropyridine 3-carbaldehyde with substituent at 5 and 6 position. So this is the invention what we have it. If you look at it carefully here, there several bonds are forming and then several bonds are breaking. That is the beauty of this reaction, the complexity of the reaction. In spite of that also, we were able to get good yields. The next thing, once we prepared that aldehyde, we thought that, we should look into the Bailey-Sielmann reaction. That's what we started. What is Bailey-Sielmann reaction? Very simple. You have an aldehyde and an electron withdrawing group carrying olefin, either acrylate or acrylonitrile, a tertiary amine, a catalyst. You put them together and stir it, you get this Bailey-Sielmann adduct, a highly densely functionalized molecule, what you get it. XH will be, if it is aldehyde, hydroxy group, electron withdrawing maybe methyl ester or this olefin, and this also aromatic moiety. So you do have several, groups which are very reactive, and then you can use them for further functionalization. That is the organic transformations you can conduct it. That's why these things, Bailey-Sielmann adducts are become famous, because the further transformation, chemical transformation becomes, easy, and you have several groups attached with that. So what is the mechanism likely? You do have DAPCO, that is the tertiary amine. It first reacts with the methyl acrylate. You do have some kind of a zwitterion. This zwitterion reacts with the benzaldehyde to get this another zwitterion. And then this zwitterion, it gets converted into an O minus, and that O minus picks up a proton and then removes this DAPCO, a tertiary amine. So these are all the equilibration conditions. So it depends upon the solvent and then other conditions also, polarizability and so many things are involved in it. But this is the standard way of representing the Bailey-Sielmann reaction. This is the basics of Bailey-Sielmann reaction. So next what we did, we prepared this aldehyde, pyridine carbaldehyde, and then we tried to make the Bailey-Sielmann adducts out of this. So if you look at into the data available, so in aldehyde and then methyl acrylate, DAPCO, it takes something like 7 days, 9 days, 13 weeks, 20 hours and 48 hours. Okay. And then the other reaction also, if you take it 2 days, 4 days, 18 hours and 20 days, and the yields are also okay, fine. If you analyze this, all these Bailey-Sielmann reaction takes enormous amount of time. So in our system, what we found out is you take the pyridine carbaldehyde and the acrylonitrile and DAPCO, methanol, and then you add it at room temperature a little bit. Immediately you have a reaction and you have a 99% yield of this. 24 hours it took if we add the acetonitrile as a solvent. Okay. So 1 equivalent DAP. It took 65% in 4 hours. Whereas we removed the solvent and then we did the neat reaction. So in about 4 to 5 minutes, we got this. So it is the question of addition of your aldehyde is the delay. So what is the explanation we offered is, the dielectric constant and polarity of the medium, which is responsible for giving this kind of a high yield and forward reaction going faster. So we prepared several whole bunch of 2022 pyridine carbaldehyde involved Sielmann adducts. And then next, what we looked into that, quite a few people they published results mentioning that the Sielmann adducts involving this ketone, cyclic enone, cyclopentanone, and cyclohexenone found to be unstable. That's what they reported. And what we did is we prepared this and then took the X-ray diagram for this, and then a whole bunch of, again, we make all kinds of derivatives involving this cyclopentanone and cyclohexenone, and we showed that they are perfectly stable, absolutely no problem about it. Maybe yields are a little bit lesser. That's all what we can say. What they did it. In our case, it is giving a fantastic yield, 85 to 98% yield. That's what I would say. That means there's no problem in making these Sielmann adducts involving cyclopentanone and cyclohexenone. Then what we did is we chlorinated it, because this chlorinated and brominated Sielmann adducts are another important intermediate to take forward in further reactions, further chemical transformations. So we prepared over 22 derivatives and then studied this chlorination reaction using trifosine, pyridine, DCM. And then you do have immediately you got converted into this chloro derivative. Okay, this we published in Journal of Chemical Sciences. So the beauty of this reaction is, you can purify them without much trouble, because this is a clean and green reaction, whatever. You dump it in water, you get it in this, and then you collect it. That's what. And an interesting thing is about the stereochemistry of it, which you will see in next slide. So we prepared all these things. So when you have an ester as an electron withdrawing group in the Sielmann adduct, the chlorine goes into this, and then you get a trans product. So when you use a nitrile, you get the cis derivative. That is the interesting thing. So this earlier also people have explained this based on the mechanism, some mechanism they proposed, and then they said nitrile is giving this, and then ester is giving the other stereochemistry with the associated with the double bond. That is what. So this is what's important, because we are going to use all these chlorides or bromides in a further chemical transformations involving these Sielmann adducts. So then what we did is we prepared all these Sielmann adducts involving pyridines and then aldehydes and all. So many we have prepared, and we published in Bioorganic Medicinal Chemistry letters. So what we did here is we looked into the anti-malarial activity. This we collaborated with the Malarial Research Institute, New Delhi. So we took this chemical and treated with that. So the IC50 values are given here. We compared with the chloroquine. So when you compare with the chloroquine, you do have that much, not that reactive. But if the person is resistant to this chloroquine, then this will be useful to administer it and then go for a whatever therapeutic use. That is what we realized. So at the two stages we have analyzed it. It is Shigant maturation inhibition or the total parasite growth inhibition. 2 points which we have looked into that when we use our compound. This is the Shukla Biswas. Dr. Shukla Biswas was the lady who was there at Malayalam Research Center, New Delhi. She helped us. And we published a couple of papers also, even after her retirement. Yeah. The another thing what we did is we took the amino, acetonylide, and then used the same Wilsmeyer-Hack reaction and prepared various phenolines. chloroquinoline carbaldehyde and then converted them into a Baylis-Hillman adduct and then studied the anti-malarial activity of these Baylis-Hillman adduct. So, here, compared to chloroquine, it's not that great, but it's okay. 2.5 or something like that. We are close to that. 1.8, we have it. So maybe, this is in micromole concentrations. So it is not that great reactivity, but nevertheless we tried and then we looked into that. So as Pawan Tagapur says, we were able to patent it also further. This we published in Heterocyclic Communications. The next slide is about the pyridone synthesis. Then what we did, we started making pyridones. So this is what a simple reaction. Baylis-Hillman adduct, you have an hydroxy group. Convert that into acetyl derivative, O-acetyl derivative, acetyl chloride or triethylamine or pyridine, you get this. And then this acetate, Baylis-Hillman acetate is treated with enamino ester, which is here in this below it is shown, enamino ester. When you mix these two and conduct a reaction, you get the pyridone derivatives. Okay, it is tri-substituted pyridones. Various substituents are there. You can do the same reaction with an aliphatic instead of aromatic aldehyde. You can do it with aliphatic aldehydes also, but you get the aliphatic substituents attached with the pyridone moiety. So if you look at into our all research, the concept is scaffold based or the structure based. So you can see it always it is scaffold based and structure based thing. The earlier slide where you have the quinolines, they are the moieties useful for anti-malarial activity and antibacterial activity also. That is the structure based and scaffold based it goes in. Okay. This we published in tetrahedron letters. So whereas if you take the nitrile instead of ester, you get aminopyridines that we published in synthesis. What is the mechanism? Very simple. You take the enamino ester and then treat with sodium hydride. You generate anion, N minus, and then this will be, a nucleophile of different, okay, 3 types of nucleophiles are equally probable. This C minus, it attacks the, this double bond and then eject the acetate, and there it starts. This is the carbon-carbon bond formation, and then followed by quickly, cyclization and then proton migration gives you the pyridone moiety finally. So when we prepared that, we published this with an idea that these pyridones have the biological activity as a cardiotonic agent. So if anyone has a heart attack, these pyridones will definitely help him to recover from the heart attack. That was the objective. So we started working on it. This is almost a 11 to 12 years of our research saga. We have been working on this to pull that because all these things will be slow and you require to be consistent and then keep motivating yourself to take it to a stage where people can appreciate it. So these are the variety of pyridones which we have prepared, and then we have the US patent and Indian patent because these have the medicinal value as a cardiotonic agent or the PDE3 inhibitors, phosphodiesterase inhibitors, which I will tell you. So how do you make the enamino nitrile? Why I'm showing this slide is there are so many assistant professors and then senior lecturers all are attending this webinar. So you can also plan this. This is a very, very simple reagent and then chemicals which have been using it. So in acetonitrile should be available. Any other nitrile you take it and treat with potassium peroxide, you get enamino nitrile. Okay. You take the ethyl acetate and then treat with liquor ammonia, you get the enamino ester. You can change this methyl group to some other, and then you get variety of enamino esters pumping. It's the same thing here also. If you change the R group associated with the nitrite, so these are all you can make it in gram quantities. Okay, that is the advantage of these chemistry. Very simple and then chemistry. So aminonicotinates, how they are formed. This is also another thing. This we published in 2010, which we have not used them for for any activity, which we are going to do it sooner or later. Now coming to the biological activity, if you look at into the literature or the drugs available in the market. The milrinone is a drug available in the market. Amrinone is available in the market. Silastazole is available in the market. You can go and buy it in a medical store. Silastamide is also available. Whereas the other thing, they're all still in the stages of introducing into the market. But these 4 drugs are available. You can buy them. If you look at into these 4 drugs, the common point what you have is the pyridone. Pyridone moiety is the common. That is the structure or the pyridone scaffold or pyridone structure is a common feature in this, the drugs what we are, listed here, 4 drugs which are in the market. The others are also have some, but not that much. Okay. The next slide, there's some more thing, drugs which are already in the phasal, clinical trial are being listed. So what is this phosphodiesterase? It's a phosphodiesterase is an enzyme which hydrolyzes the phosphate group, okay, and then convert that into a ATP. If you have ATP, it hydrolyzes that and convert into inorganic phosphate. That is what it does. Okay, so coming to the little bit more biology, cardiovascular actions of cyclic AMP dependent PDE3A inhibitors. So what it does, PDE3A, it recognizes cyclic AMP or cyclic GMP and hydrolyzes that into a phosphate group. So what is that it has? It has a vasodilation, enzymic vascular resistance, decreased arterial pressure, increased organ perfusion. There are so many functions it has. So this PDE3 dependent, cyclic AMP dependent PDE3 inhibitors. Coming to the real point now. So these are the PDE3 enzymes. You do have a PDE3A 1, 2, and 3. 3 proteins are here, 3 enzymes, and you have a PDE3B. Where are they located? They are in the heart, vascular smooth muscle, platelets, oocyte, kidney, cervical. The PDE3B is also there. Hepatitis, kidneys, developing sperm, lymphocytes, macrophages. This is the localization of it. So these are the protein. The structure is being elucidated. Given, and then the proteins are also enzymes also isolated. You can isolate them from the blood and then assay them. So what is the thing now? The thing comes here. In humans, you do have the sympathetic nervous system which secretes the NE, that is norepinephrine. It's a hormone. Okay, that hormone activates the beta part of the beta segment of the G protein. With beta segment, it goes on binds with the adenyl cyclase, that is another enzyme. Okay, and then that gets activated. This adenyl cyclase, what it does, it reacts with ATP and converts the ATP into cyclic AMP. This cyclic AMP will phosphorylate the heart muscles or the artery, whatever walls, or it controls the platelet activation. So here, this is the cardiac muscle which phosphorylates cyclic AMP, and then cardiac muscles get the energy, and then it will contract. So it will start functioning. If it is intervened by PDE3A, then you'll have a hydrolysis giving you 5 AMP. So it is an energy wastage process. Okay. If this dominates, then you have a problem of heart attack because energy is not supplied to the heart muscle. Therefore, the heart will be weak and it may not squeeze that much. That is the thing. And then coming to vascular smooth muscles, okay, where you do have a phosphorylated myosin and then which leads to contraction, a relaxation occurs in this artery. So it is required. If it is intervened by PDE3A, then it gets into hydrolysis and then you get cyclic AMP, energy wastage process. Comes to the platelet. So the next slide will give you more pictorial representation. You can appreciate it. This is the heart and this is the heart muscles. What you have seen it is a sympathetic nerve. So it secretes the norepinephrine, adrenaline, right? Adrenaline binds with the beta segment of the G protein, which activates the adenyl cyclase, and then ATP converts to cyclic AMP and all these things. So this cyclic AMP is involved in phosphorylating the heart muscle and then energizing it, and then heart function is possible. Suppose if PDE3A involves, and then that will hydrolyze cyclic AMP to cyclic AMP. What we can do, the best thing what we can do is you make a chemical and then put that chemical which binds with the PDE3, that is the phosphodiesterase 3A, and then stops hydrolyzing cyclic AMP. If cyclic AMP is available, heart will start functioning. So if any person who has, imbalance in their heart functioning, heart muscles are paralyzed, then you administer this ambrinon and milrinone or my chemical, then it should be cured. That is the objective. I hope you people can appreciate my efforts in speaking to you the biology or, medicine. The next one is artery. In Telugu, they are called dhamanulu. Okay. So you do have arteries are always thick. The other thing is siralu and there will be thin kind of thing. And then this takes the blood along with the nutrients and oxygen. So the arteries muscle, where you have a vascular smooth muscle, that also gets phosphorylated. If you inhibit the phosphodiesterase at this stage, you get, this energy energized, and then your artery, gets activated and gets relaxed because you are supplying enough of energy. That is the objective. This is the therapeutic point, and this is the therapeutic whatever direction what we are discussing. Let's get into the third one. Third one is the PDE3, role in platelets. This is the biggest problem. Over 90, 95, 96% of the people, they will have a problem. I also had the same problem. So at the age of 52, 53, I had the platelet aggregation suddenly. So I was hospitalized and I was given a streptokinase injection where these platelets gets deaggregated or solubilized of this deaggregation. What is the reason for this aggregation? It is the same thing. If cyclic AMP is not available, then these platelets gets activated and gets aggregated and it forms some kind of a thrombus. The medical professional, they call it a thrombus. Thrombus means ఒక జిగురు లాగా ఉంటుంది. ఈ చింతపండు జిగురు లేక బెల్లం జిగురు చూసాం కదా, అలా ఉంటుంది. అలా ఏంటి అవుతుంది, ఎక్కడ ఉంటుంది, ఈ ధమనులు ఆర్టరీస్ లో తయారవుతుంది. Once that kind of a thrombus is filled in the arteries, then the blood flow will not be normal. It will be decreased. When the blood flow is decreased, oxygen supply will be cut off. Okay. When the energy is not available, these, further this aggregation of this goes on. It gets multiplied. It's a chain reaction. It becomes, and then you have a problem of blocking the artery. This is the kind of a thing which many people are suffering in India and all over the world. So this is one of the strong reasons. So if you administer my chemical or the pyridone, that is ambrinon and milrinone, they stop this PDE3A, inhibit the PDE3A, and then make available cyclic AMP, provide energy which uses these platelets and gets deaggregated. That means a solubilization process is taking place. This is the therapeutic kind of thing. So what we did with our chemical is the next slide, you can have it. So ambrinon and milrinone are the two examples. This is the standards which we have used. The milrinone is the standard if you look at it. And then we have used all our compounds, whatever prepared, something like A to Z, okay? And then in that, these peaks, they respond very high. They show percent of inhibition very high. Then what we did is, we calculated the IC50 values also. And then what we did is, we approached a computational chemist who pulled out the protein structure from the website. And then what he did is, he did docking studies. Docking studies, what I understand is, you put the original molecule like ambrinon or milrinone in this, and then take the numbers generated by computer, and then you remove this ambrinon and milrinone, put all the new compounds what we have prepared, and then look at it, the numbers. So that is called docking. The score what we get is pretty good. That is, the score is better than the standards drugs marketed, that is ambrinon and milrinone. And then if you look at into this table also, we have plenty of chemicals showing a IC50 value much better, and then very excellent numbers what we got it. So this gave us confidence that we should go further and then study it. After publishing the tetrahedron letters in 2009, we took almost 3 years. So one must be patient with this research. Okay. After 3 years, we were able to publish bioorganic medicinal chemistry letters. That is the journal we opted for it. So it deals with chemistry as well as medicinal chemistry and biology. So next slide, is the background. So in that, so many compounds what we have prepared, in that these 5 compounds are found to be better. If you look at it, the IC50 values, 1.68, that is the 8V, 2.66, 8J, and then 8.47, 80, 7.49 HC, and 7.38L. Okay, so these are the standard things: screening and 2-period derivative for PD3 enzyme reaction. So we selected these 5 and then took further. Okay, so in European Union and all, they use silastazole as a drug to control this, blocking or ischemic stroke, hemorrhagic events and all. This is the famous drug, silastazole. Okay, and then therefore silastazole could be used in prevention of stroke in patients with non-cardioembolic stroke. So it inhibits the PDE3, no doubt about. What you have is the platelet aggregation, vasodilation, all kinds of things. So what we had to do is, when we go for an animal study, this is here I'm starting the animal studies what we have conducted in collaboration with the German medical professionals, reduces ischemic brain damage, neuro and endothelial protective blood-brain barrier, protects against the hemorrhagic transformation after transient middle cerebral arterial occlusion, anti-inflammatory, inhibition of platelet aggregation. So why should we go for new PD3 inhibitor? Specificity and lesser side effects, therapeutic efficacy and mechanism of new PD3 inhibitors after stroke. This is the objective in making all those things for PD3 inhibition. Then what we did, before that, I missed a slide. I should tell you, the German professors, what they did is they wrote a report, and then they mentioned in that report that there is something like 260,000 stroke patients or heart attack patients, in Germany each year. So there's a huge amount of this. I think this number is less than the present corona what the Germany faced. It is only a small country, Germany. It is as good as combined, 2 Telugu states like Andhra and Telangana. If we combine, probably that may be the state, But humans wise, the population wise, it is far less than these 2 combined states. These are the 2 medical professionals, Peter Kraft and Michael Biber at University of Würzburg Medical School, Germany. They are doctors. Okay, this is some report, and they say that this is a very severe in Germany, and then one has to improve our medicinal aspects on this to save the people. Okay, so what they selected is the mice, that is the rat. Mice or small rat and whatever. So they took the brain of the rat. It looks like this, and then this is the cross section of the brain. These red color things are the arteries taking blood to the various part of the brain. This is what I said. And then here, if you come it, there are labeled. In here, it is MCA, which is colored here. So the one of the artery is called MCA, middle cerebral artery. Okay. So what they did is they introduced one occlusion, that is the block in the middle cerebral artery of the mice or rat, small rat. Okay. So once you introduce a occlusion, what happens? The blood flow decreases, oxygen will not be available. Then that part of the brain goes off the line. That part of the brain cells will be, non-functional or close to dead. So in that condition, you start experimenting. Then the question arises, how to confirm that this part of the brain cells are dead. So what they did is they cut it, sacrifice the animal, cut it and taken out this brain after introducing a transient middle cerebral arterial occlusion. There is a block, and then they sprayed this tetrazolium chemical on it. So wherever the brain cells are of the line non-functional, it showed fluorescence. Wherever the brain cells are good, it didn't show any damage or fluorescence. That means they are good cells in the part of the brain. Another part of the brain, they are affected and non-functional. This is the assay they have developed. After developing this, introducing a arterial, block, arterial, whatever you call it, occlusion, middle cerebral arterial occlusion. They have injected silastazole. They have injected our compounds, 3 compounds, that is 8J, 8O, and 8V, and then started looking at the improvement in the animal. That is the behavioral aspect of the animal. Within a day, the animal looks to be back in action. How do you say that animal is back in action? Once again, you sacrifice it, dissect it, and take the brain part and then spray tetrazoleum and see whether that part is fluorescent or not. So there is a reduction in the fluorescence, clearly indicates that brain cells are getting repaired or recovered. That is the idea. Another thing what you can employ is the when occlusion occurs, when you introduce a block in that artery, middle cerebral artery, the animal becomes so inactive it cannot move because of the low energy, and then brain is totally off. So tail, the rat tail will be wagging continuously. That is being stopped. And then rat limbs, which have a good grip, that grip also lost. And then rat, the movement of that, mouth movement will be different. That is also stopped. You can make use of these 3 behavioral aspects. Recovery in these 3 behavioral aspects confirms that the chemical injected, the chemical prepared by us, which is pyridone moiety injected, is working and solubilizing the clot introduced artificially in middle cerebral artery of the rat. That is what the conclusion which we make it. So what is the outlook, what we are going to do next? This is called animal model. Still now what we did is we prepared them, tetrahedron letters paper 2009, and studied the, what is that, PDE3 inhibition 2012, and then we get into the animal model, that is 2018 we published paper. So it took almost 10 to 11 years. Now it is 14 years we took time to get into that. What we achieved, we were able to identify that pyridone moiety is very good for these studies, and then we were able to generate hits, and then we were able to convert hits into lead molecule, and then we were able to see the lead molecules getting into the phase 1 trials. So during this process, what we got tetrahedron letters paper, synthesis paper, bioorganic metal chemistry letter paper, stroke AHA journal 2018. This is, we published it with the German doctor. And then we have 3 inventions, 3 patents we have filed. And then, what we have prepared? We have prepared over 600 chemicals during this way. And then something like 4 PhDs we were able to, collect it. 4 PhDs are given. So what we had to do further, memoir analysis, magnetic resonance analysis of the bleeding complications, and then effect of neuronal apoptosis, evaluation of platelet function, dose dependency. Now from the animal studies, it's clearly confirmed that the chemical what we have prepared is working, and then clearing the thrombus or the block which is prepared. That is, it affects the solubilization of the block. This is the story. 2 pyridone derivatives. What we did during the other process, we were able to make the dihydropyridones, and then dihydropyridones converted photochemically, or using silic ammonium nitrate also, we can convert that into pyridone. Another method we pumped in, that is in the photochem photobiology 2009. So students required some publication, so we had to work for that also. So these are the several pyridones which we prepared from this route. The next thing what I'm trying to speak about is about the aldose reductase. This is about the sugar problems who has it will be there. So what is the sugar problem? When you improve your glucose concentration in your blood, what happens there is after a certain concentration level, the NADPH enzyme comes into picture, converts the glucose into sorbitol, involving aldose reductase enzyme. Okay, so once the sorbitol comes, this will have a different property. It will have a lesser blood-brain barrier, and it gets into the brain. Once it gets into the brain, then your nervous system is collapsed. So one has to be careful. That means at a level where the glucose is very, very high, then you have to administer this drug. What is the drug? The drug is called epelristat. This structure is given in this slide. So how do we make the epelristat? Standard formula. We are using Bailey-Sielmann chemistry. I will show you. So what we did here is the inhibition is an important therapeutic strategy. Is only the approved drug. Okay, that is epelristat is only the approved drug. Then there is a necessity to go for a new drugs having better efficacy and less side effects. These are the things which we kept in. Those are the objectives, and then with that objectives we move forward now. So these are the epelristat. The other things have been tried and kept it in abeyance. These are all the drugs which are in the except epelristat. The other structures, they're all in the clinical trials. We say it. They are kept it aside. That's all. Only the epelristat is the drug available for this reductase inhibitors. So how do you make it? You have the rhodonine derivatives. Hello. These are the rhodonine derivatives which we have shown, and then from that you can make series 1, series 2, series 3, and then series 4 as it's shown here. So we were able to make around 44 compounds. What is the chemistry? Very simple. You make the Bailey-Sielmann adduct and then convert that into a aldehyde using rearrangement, allylic rearrangement followed by oxidation. You get the aldehyde, and then condense with the aldehyde this. So 1, 2, 3 steps, you get the final compound. That is the thing, which is very, very, the chemicals involved is also, once again I say, they are very, very simple, and then you can make them very quickly, right? So you have an naphthalene derivative leading to that. You have pyrrole, thiophene, furan, all kinds of things which we have prepared. These are the whole bunch of chemicals which we have prepared out of this Bailey-Sielmann adducts, which is having close to epelristat structure or epelristat analogs, you can call it. Then what we did the docking studies also. The docking study is becoming important, and then the students also get fascinated with the computation techniques. They are very, very eager, hungry to learn the computational technique. It is better they can learn and then sit in the computational room and then do something. That is what we encouraged it. And then what we did, we assayed for activity, all those reductase activity, and we published it in European Journal of Medicinal Chemistry. We do have something like 1, 2, 3, 4, 5 compounds which are, one is better than epelristat, and then the remaining are all close to epelristat IC50 values. So we do able to generate hits and leads. At this stage, we stopped it because we already, engaged with the cardiotonic agent and took them to the animal model where, these we cannot take it. So we kept it aside, and then we just published it and patented it. That's what happened. And then next thing is the tetrazole. If you look at the tetrazole structure, these are sartans are there, losartan, ibrasartan, and valsartan. They're all, controlling the blood pressure. These are sartans. So the tetrazole moiety is the most important in this sartans. Okay. So how do you make it? You make the Bailey-Sielmann adduct and convert that into acetate, and then treat with sodium azide. They're all cheap chemical. You get the azide, and then reduce it. You get the amino derivative. Use it immediately, amino derivative with formaldehyde and then another sodium azide. You get the tetrazole. You get it. Okay. So if you use nitrile, you have a one stereochemistry, and then if you use ester, you have a different stereochemistry. That's what the sequence is given here. You can make the whole bunch of tetrazoles which we cannot make it. So what we did this tetrazole, we looked into the anti-cancer activity, and then there are quite a few things were found to be at the micromolar range. And then how do we assay it for anti-cancer activity? There are enzymes and then cells, online cells, they're all, they all did it, and we published it also. The another assay what we did here is very interestingly, we got this crystal structure for these two tetrazoles also. We published it in Medicinal Chemistry Communications. Now the name of the journal is changed. So when you take the DNA, that is the CD, calf thymus DNA, and then add our chemical, there is a change in the absorption as you increase the concentration, decrease in the absorption. Whereas if you take the same chemical, okay, and then add it to that and look at the luminescent property, there is an increase in the fluorescence. So what we were able to say is the added molecule, whatever the tetrazole, binds with the DNA, and then when it binds with the DNA, the mobility of the molecule is reduced. That's why it is showing higher fluorescence. So this confirms that added molecule has an affinity to bind with the DNA. The next one is, synthesis of tetrazolones. So these are the tetrazolone. It has an hybrid, herbicidal activity, obesity treatment, and also beta adrenergic receptor. All these things. It has a role as a. These are all the chemicals which are available in the market. How do you make it? Very simple. You make the Bailey-Sielman adduct, convert that into a Bailey-Sielman bromide, and then you take isocyanate and then use trimethyl silyl azide, and then you get the tetrazolone. You do have this NH proton that you treat with base N minus, and then coupling takes place here, and then you do have variety of the tetrazolone moiety is prepared. The next slide is the tetrazolones, number of tetrazolones what we prepared as usual, and then we tried this antibacterial and antifungal activity. The standards what we use is ciprofloxacin or the itraconazole, that is the antifungal agent, and then that we were able to get that there is some activity is there. We had to improve with the structure. What we realized is we had to improve the structure and then make it more active. That is the next part of it. We had to try. So the next thing is the quinolines which we have prepared. Quinolines are also famous. And quinoline antibiotics are already in the market. That is ciprofloxacin, all kinds of fluoxacins you have it. So this also has an importance. So we prepared this, quinolines, okay, and then we studied the antibacterial activity, against penicillin and streptomycin. So these compounds shown to be, very good. You can see the numbers. Quite a few things are very good. We patented it also. And then this paper is cited over 200 times. Many people are reading it and citing. That is the thing what we look into that. This is about the antifungal activity. Published it in Bioorganic Medicinal Chemistry 2006. Okay. And next slide is the how we prepared these quinolines. Okay. Bailey-Sielman acetate, that standard, and then that is treated with nitroethane, so it gets under cyclization. So it always 2 steps or 3 steps to the target molecule that we put it. That is the ease of chemistry, ease of doing chemistry. So that is what we derive. The other things are also like that. You do have a tri-substitute, tricyclic quinoline. We have tricyclic structures also involving quinoline moiety. This is the mechanism. What nitroethane, you treat with potassium carbonate, you have an anion. Anion, it replaces this acetate, okay, in a allylic way. And then again, another anion is generated. It replaces the chloro and makes carbon-carbon bond. And then loss of this nitrous acid will give you finally the quinoline moiety. Based on the different substituents, you can vary the substituent and make various analogs of it. This is the naphthyridine synthesis. Further, we got into that. And then you have 2 nitrogens side by side. So many, analogs also we prepared. We published in Helvetica Chemica Acta. Once again, it involves the Bailey-Sielman chemistry. So Bailey-Sielman acetate is treated with tosilamine or ammonium acetate or, sodium azide. After sodium azide, you treat with triphenyl phosphine and water. You get the naphthyridine. So, see, the beauty of this is you can make them with very simple chemicals. Once again, I say, you require only a clear planning, a clear, whatever, motivation is required. You can make all kinds of things, even in your laboratory also, your practical laboratory, MSc practical labs, you can make all these things. That's what I tell you. Yeah, Bailey's. The mechanism is given here. So tosilamine, when you treat with potassium carbonate, you have N minus and it gives, and then again it replaces this, okay, and then losing the tosic acid, you get the naphthyridine. You can use the DAPCO also to create and then facilitate the reaction because you get this kind of a cation, so it becomes a better leaving group for azide to get in. Otherwise, it undergoes side reactions also. To control the side reactions, we have used DAPCO to make the, this kind of a cation. And then treated with the triphenyl phosphine, you do have this kind of intermediate, N double bond P, and then when you dump it in water, you get triphenyl phosphine oxide out, and then you do have an NH2. That NH2 intramolecularly, displaces chlorine and forms the carbon nitrogen bond to get the naphthyridine, dihydronaphthyridine, which during the column, silica gel column chromatography, it loses another hydrogen in the presence of oxygen. Okay, it pulls out the two hydrogen, and you get the finally naphthyridine. That's what we see. Mechanism, based on the other things also. This is the ammonium acetate. So in this ammonium acetate, what you have, ammonia and the acetic acid, they are in equilibrium, okay, in anhydrous methanol. So the ammonia which reacts with this, and then you do have it, NH2 loses proton, and then this NH2 replaces this and forms a carbon nitrogen bond, and then aerial oxidation gives you naphthyridine. You can use the benzylamine, triethylamine, where you can block this NH group, and then you can make the dihydro naphthyridines also. This also we prepared to prove the mechanism. That is also we did it. Yeah. Again, flunarizine and cynarazine derivatives and anti-migraine drugs. These are the flunarizine and cynarazine. They are used, they are available in the market. You can buy them when you have a migraine, that is a headache, which migrates all over your brain. That is what happens. These are the pictures it has given how the migraine happens and other thing. These are the chemistry is very simple. You make the Bailey-Sielmann product, and then you make the Bailey-Sielmann bromide, which is in 2 steps. You can make it, and then you prepare this derivative, okay, and then couple it with this. You got the flunarizine or cynarazine. It's very simple. So what we are doing, we are replacing this and this. So we are making variety of analogs to test them for further activity. We published it in medicinal chemistry later, but we could not find the right collaborator to do this. By the time, the student also finished her PhD and then published it, left. So during this time, what happened, we were able to help the industry also. These industry people, maybe same thing in, Visakhapatnam also, probably making these 2 derivatives. When they send the consignment to the foreign, okay, the foreigner, receiver, what he raised an objection that the stereochemistry of the double bond is not right. Then what we did, the people, they came to us, they said they need it in 1 day. I said, no problem. So I just took this cynarazine in an NMR tube and irradiated it. So it got equilibrated into this, almost 50-50, and recorded the NMR, and then recorded the HPLC to show 2 isomers. So that NMR and that thing, they faxed it, and then they were able to get the consent, clarified, and then they went without any problem. This kind of things also we did, on several occasions in vitamin D and vitamin A and all these, double bonds also we do it to the industry. So cynarazine is used for treating nausea, cerebral epilepsy, antihistamine, calcium channel blocker. There are so many functions which we have it. Okay. This is once again, we tried this, photochemical photoisomerization of the double bond, and then we published it in European Journal of Medical Chemistry. Our photochemistry knowledge we do have, so with that we were able to generate both the stereoisomer, and then that's how we published it. The conversion is 20% when you don't use sensitizer. When you use the sensitizer, you do have a higher, Z geometry or cis geometry, you have it, and then we studied their antibacterial and then antifungal activities. So now I thank you all for your kind patience, and then I thank all my students listed here. Quite a few master students are also listed, research associates are also listed. These are the guys to be, completed. And then I thank my director, Dr. S. Chandrasekhar, for the, encouragement and all. These are the students who are all, associated with me. This is the another photograph. This is the another, and this is the last batch of students who are associated with me. Now there are quite a few are left. She is Parvatineni, Mrs. She's went to USA as a Fulbright fellow, and she is looking for a job. That means a suitable position, suitable job she is looking. And then she is Dr. Mohanty. Her son came to intermediate, so she gave up postdoctoral fellowship with me and then stayed at home. She went to Germany as well as UK, and now she got a job in Novartis, and she just finished the PhD 1 year back. This guy is Yedkondalavadu, Tirupati, Namo Venkatesha. He is at ISER Tirupati as a technical officer, and then he's just submitted he got the PhD just a few days back, and then he's looking at Taiwan. And then this guy is Harsha, who worked at NIA, National Investigation. He is known as Bejawada Harsha. And then he's going back to he has submitted the thesis, hopefully he'll getting. This guy is Bhikshapati, he's Vorgalu, Warangal Raja. He left to Korea, he's working in Korea as a postdoc. He's Appalaidu, he's from Vijayanagaram. And then he is also, nice guy, very nice guy. He's put on a lot of experience in drug. He's going to complete the thesis. He's Narendra Reddy, who has done a lot of work for me. He is still a faculty under whatever, and he's also looking for a job. He's Raju, who has links with Adikavi Nannaya University, and then he is trying to submit his thesis soon. He's Bharat, who is in France now. He is coming back because of the COVID problem. He's Jagadish, who is at Naipar Guwahati. Nagaraju, Pithani Nagaraju, he's from Guntur, and then he is now in Korea. He is also Ramesh, my student, who is working as a technical officer. Lakshmi Reddy is writing his thesis, and then he's also started writing the thesis, Uma Shankar. He is my teacher, PhD teacher, Ramamurthy, and then this is the group. Quite a few are left, only 7 people are living, and I thank you for your kind, attention and cooperation. If there are any questions, I'll be very happy to answer. Thank you. Hello?
Hello. Thank you, sir. Sir, participants, please, any questions?
Hello?
You can ask.
A little bit loudly, please.
Sir, this is Vidyasagar, sir.
Babu, cheppu, అయిపోయిందన్న lecture.
Okay, sir. Okay.
Any questions? అందరూ ఆకలిగా ఉన్నారేమో 1:30 అయింది. I got it.
Thank you, sir. Thank you, sir, for your valuable lecture. All attendees, if you have any questions, please ask. Please unmute yourself and ask questions.
సదస్సులందరికీ నమస్కారం అమ్మా. ఆకలిగా ఉన్నది అయినా ఏం పర్వాలేదు, మరి అడగండి, చెప్తాను ఇంకొక 5 నిమిషాల్లో ఏదైనా ఉంటే. The interesting part of this lecture is you can plan in a very simple way. You cannot say that I don't have this big chemical and all kinds of things. And nowadays NMRs, you can get the NMR by paying 100 rupees or 200 rupees, something like that. Mass spec also you can. So you can link up with some people, biologists or pharmacy. You can initiate a small research. You can publish small papers. Absolutely no problem. That is what I feel. It should be possible with you all. Yes, please. Any question?
Sir, this Rashekar Reddy, sir.
Oh, yeah. Good morning.
Good morning, sir. How are you, sir?
Great, great. Tell me.
Sir, that each isomers you you did some heating and all. Could you please elaborate on that, sir? That's why I could not get you.
We have published a chapter on EZ isomerization. Okay. So when you take the drug molecule, particularly the synergine and all, okay. So what happens if it is a cis isomer, it is inactive or less active compared to trans isomer, E isomer. So it becomes essential to establish that what you have synthesized is the trans isomer, and you have to prove rigorously that is the involved. When they send the consignment abroad, they claim, no, it may be the cis isomer. We do not know. How do you prove? That when they asked that question, they came to me and I was able to solve it within no time. Like that, small tricks we play using photochemistry, particularly EZ isomerization. There are so many drugs which have this EZ isomerization possibility, and then they take that as an impurity also. Many a time they demand. So impurity research is also very intensive in this Visakhapatnam as well as Hyderabad. So one has to make it and then submit the impurities up to something like 20 grams or 50 milligrams, 20 milligrams or 50 milligram quantities. That is what happens. So it has a lot of importance, practical application and practical importance we have it.
So I just want to curious to know, sir, is it like mostly the trans isomers only bioactive? Is the general statement, or cisandro isomers are not much biologically active?
Okay, I will tell you now. See, we have pigment in our eye, rhodopsin pigment.
Okay.
Okay, there 11 cis retinol is the pigment. So cis isomer is also there. Okay, so it has a biological activity. Every day our eyes, getting converted 11 cis to all trans, and then another isomerization enzyme comes that converts the all trans to 11 cis. Okay, and then our BV Subbareddy, who will be probably my division. Now I am his division, is the division head, who will be speaking, I hope, on pheromones. Most of the pheromones, they have a Z geometry, and then he will further speak about the importance of Z geometry. That is the pheromones which attract these insects and then trap them. That is the methodology he has developed. Our BV Subbareddy, who is the Dr. BV Subbareddy, who will be one of the speakers also. So it is not like that. Only all trans geometry is as important. Cis geometry also is important. Nature has picked up many things. Okay, like rings, you take it, trans ring junction versus cis ring junction. Some steroids will have trans and some steroids will have a cis ring junction. So it depends upon the nature how it develops. That is what I want to say.
Thank you, sir. Thank you for your wonderful and informative lecture, sir.
Hello.
Sir, thank you, sir. Thank you.
Okay, then I think you are all hungry. Looks like. Okay, there was no question from the
Yes.
సదస్సులు ఏమి మాట్లాడడం లేదు. So you can talk to me also anytime. I'll be very happy to answer your questions.
Okay, sir.
Okay.
Okay, sir. Thank you, sir.
Thank you, Jagannath Reddy garu. Thank you very much.
Yes.
Right. Bye bye.
So with this, today's session is completed. The feedback form will be given under WhatsApp group. And again, we will meet tomorrow at 10 o'clock for tomorrow's program. So thank you, Anandal. That's all for today.
Okay.
Thank you, sir.
Thank you.